you. I'll be moderating and first speaker today, so you'll get a lot of me. But before we start, Steve's got a few comments about today, and then I'll come up and start speaking.
Yep. Morning, everybody. I sent out an email, and I'm not sure everybody probably got it just because of where we are. We know the weather's coming. There's going to be a pretty good storm system coming through this afternoon. We spoke with the hotel. If anyone wants to simply add a night and extend their stay, they're welcome to. If you've already checked out, you can go ahead and get check back in and get the room rate for the conference. You know, if you have to go, we understand. We don't want anybody risking anything, but we don't want you getting stuck on the road in traffic in a storm either. So whatever decision you make, if you're on our travel, just let me know, and then we'll go ahead and make the adjustments later on. So if you have any questions, if you need anything, if you have a problem with the hotel or not quite sure what to do, just ask. We can see if we can help. We have the YouTube is streaming live now. This is session 3. At noon, it will end, and we will go to session 4. And at that point, we'll have the breakouts, and then we'll come back for a general discussion as a big group after 3. So the YouTube folks will have some dead air time from 1:30 until 3 while we're all in different small groups. And after that, we'll be back in full strength. Everything good, Joe? This one?
I think
the one with the P's on it? Okay, I'll bring it to you. That's it, Mike. I thank everybody. Yesterday was great. Today, we're going to have lunch. We're going to do a head count here in just a little bit. I'm assuming some more people are going to come in, but we don't want them to cook for more than what we have. So if you see me standing up and pointing, I'm not pointing at you. I'm just counting you. It's all good. Oh, and if you didn't hear, the Mobile Tunnel is underwater. It's always underwater. There's a river over it. Don't slap me.
Somebody slap him. Thank you, Steve. Wait till the slides get loaded here. I think that's the next one. You ready to go, Landis? Looks like you're okay. Okay, so briefly, today's morning session is going to be on movement and migration dynamics of Southern Flounder. So yesterday we kind of, in the life history session, we touched on this a little bit, and I think it was a good foundation for what we're going to talk about today. And so what I'd like to do in the time that I have today is kind of broadly cover the migration and movement behaviors of Southern Flounder without hopefully stealing anybody's material for the talks that are going to come afterwards, but hopefully set the stage for some of the questions and data gaps that I think exist with Southern Flounder movement. So we've talked a lot about this yesterday, so I won't belabor this point, but Southern Flounder have a really unique life history. And, you know, with larvae coming into the estuary, a juvenile nursery stage in the estuary, before moving offshore during spawning migrations as adults, and then the cycle repeating. And so this is a really unique life history, which poses some management challenges. So I think a lot of times we think of Southern Flounder in the group of inshore fish with red drum and spotted sea trout. And like those species, they're managed by the states to a certain extent, but red drum are just managed during that adolescent phase to escapement. And spotted sea trout we think of as a bit more estuarine resident, although that's not completely true, I guess. But southern flounder have this estuarine coastal connectivity as part of their life cycle, where the fish are going to move offshore and outside of state waters, presumably, but we still have to manage them as a state. So what amount of interstate mixing, if any, exists is a big unknown. And so there's a potential for mixing during this offshore phase. So I'll start with early life. There's less movement information on early life, and this is actually from Nathan Fury and Jay Rooker's work in Galveston Bay, and this is actually based on abundance data. So we heard a little bit about this yesterday, that there's some dynamics during that first year of life where we may see juveniles settling in a particular part of an estuary and moving to another. I think yesterday we heard about settlement in the upper estuary. This is an example from Galveston Bay that showed settlement closer to some of the passes and actually moving during the first year up to the upper estuary. But again, this is based on fisheries independent surveys. So this is not actually movement data, but abundance data. So whether that's actually movement or just differences in mortality is not necessarily clear. But the take home there is that there can be different distributions during that first year of life and some movement potentially within the estuary. There's also been some work that we did years ago in Texas looking at fine scale movement of southern flounder during this adolescent phase. I say juvenile, it's really probably at about age 1, so at the end of that first year of life. And can you go ahead and play the animation? So this is from some acoustic telemetry work, and it's just a quick animation showing fine scale movement of 3 southern flounder within an estuarine seascape. So if you see the colored dots, those are 3 individual flounder, and the habitat there is a combination of shoal grass, which is the light green, with turtle grass patches, some sand, some oyster reef, and some salt marsh. This study was done in the winter, so these are actually fish that are about a year old that are overwintering, so they did not move offshore. And so the important thing to take home here is that that shoal grass bed is a bit senesced, so it's a mud, kind of very thin shoal grass mixture. So what you'll notice, can you play it again? Just to, I think it'll repeat maybe. The fish tend to, in this study at least, to hang on these edges of these habitats, which isn't that surprising for an ambush predator, right? So we'd see them on the boundaries of the shoal grass or the oyster reef for extended period of time. And there's also some error associated with the positioning in these tags. So it may look like they're moving around very small amounts. That's likely just the error in the tag, and they're probably positioned just outside of that habitat. So from some of this fine scale work, we were able to associate movements with temperature, and we look at this is rate of movement. So this is still movement within a pretty small sphere, right? But within that winter phase, found a strong association with temperature, where the fish were more active at higher temperatures and relatively inactive at lower temperatures in the winter. And this figure in the lower right is actually ignore the red panels. Those are actually from Red Drum. So this is from a paper that's already published. So that was in the figure. But the blue bars are for southern flounder, and that's just movement rates across different habitats. So basically more of this kind of lie in wait behavior adjacent to the turtle grass habitat, which was the main structure during that winter phase. So they're basically sitting outside these turtle grass habitats waiting for prey to come out, essentially. Interestingly, there was another study we did in the summer in this same system with a slightly different results, where the fish actually spent more of the time in the sand habitat. But if you think about the dynamics of this particular system, the shoal grass is thick in the summer. They're not bedding in the shoal grass. They actually bed in the sand, and then the edges are the shoal grass habitat. So slight differences. At a bit larger scale, these fish in the first year of life, we found some movement associations with different environmental variables, and basically that they were more likely to move greater distances. And so when I say greater distance here, I'm talking about movements of at least a kilometer. I would still say the movements were rather limited, but they made movements of greater than a kilometer in association with tidal range and temperature. So when the daily tidal range was lower, we found they were more likely to move and at greater temperatures. So again, that same patterns we saw on the really small scale in the winter, that when the temperature was very low, they tend to be less active. Now again, these are fish that are basically about a year old, and this is during the overwintering phase. But across the studies that we've done in Texas and studies in North Carolina and other areas, movement seems rather limited, at least in the estuarine phase. And there's plenty of evidence, both from conventional and electronic tagging, of fish remaining very close to the tagging location for extended periods of time. And in fact, at least in acoustic telemetry, I think flounder are a good example of a species. I think I do a lot of acoustic telemetry work, and we think about fish that we think may be dead within a grid because they repetitively ping on the same receiver over and over again. You know, you have a tag that talks every minute and a half, and the tag shows up every minute and a half for extended periods of time. That's like the flounder signal. But they're not always dead, and sometimes it takes Shane from Texas A&M, who's in the audience here today. We had a fish that was tagged that remained on the same post for 8 months. Thought it was dead, except Shane caught it like 3 or 4 times at that post during the study. So we know it was in fact alive, and eventually someone did catch it and keep it, but relatively limited movements at the smaller scale. So for adult movements, we see relatively little movement, and this is a broad generalization, during that summer phase in the estuary. Then the fall spawning migration, you know, we'll see, at least from our acoustic tag fish in the past, very little movement, and then in the fall, all of a sudden they will start shooting out towards the tidal passes. And then in the winter, they're offshore, and then a return migration in the spring. And this is kind of the general pattern that we assume is occurring. And what I'd like to talk about in the time I have remaining is kind of some of the gaps that exist in this kind of circular pattern. So that fall spawning migration, I think, is the big movement dynamic that we really think about. And this is typically in October through December. It'll vary a little bit depending on in what area we're talking about. I just use Galveston Bay here as an example. But fish will move from the upper estuary to stage in the passes in the lower estuary during this time. And this is a bit anecdotal, but those fish tend to forage pretty heavily during that staging, right before they move offshore. I think it'd be an interesting study in and of itself, but we've noticed that from tagging over the years, those fish feed very aggressively before they move offshore. And because of this very predictable movement through channels and migration corridors, and they're somewhat predictable, there's high effort and catch in the fall from some of David Smith's work he talked about yesterday, at least from Louisiana, and these peaks and fishing effort in the fall. And these are also primarily female fish that are caught. So the primary questions that I think exist, and hopefully some of the talks today will cover some of this. I think the first question is the timing of the spawning migration. So flounder are particularly vulnerable during this movement, right? A lot of the fishing effort takes place during the fall when they're moving through very small bottlenecks and choke points. And so what is the timing of this movement? What are the cues for the movement? Is it temperature? Is it photo period? Is it cold fronts? You know, we tend to see movements after these cold fronts, but I think we could learn a lot from the aquaculture research as well in this, in terms of the drivers of some of the spawning behavior and the role that photo period plays in that as well. And then how would the impact of warmer winters and weaker cold fronts change these migratory behaviors? The second question is partial migration. Do all flounder migrate? We heard a little bit about this yesterday, and I suspect we might hear more about this today. We've heard about freshwater residency, so potential for different contingents. Are there sex-specific differences in migration? Do males migrate differently than females, earlier or later? And then the potential for non-migrants, and whether that is skip spawning. If they are skip spawning, why and how often? How often do fish spawn? And then does spawning occur within the estuary? So I know from some of our work in Texas in the past, we'd show this non-migratory behavior, but it's not clear whether they're skip spawning or spawning in the estuary. And Fred talked about this a little bit yesterday, but where do flounders spawn? We don't have a lot of information on this as well. Are they going to the shelf edge? Do they spawn in nearshore areas close to the estuary of origin, or do they move with the longshore transport? And so Fred showed some of that yesterday, net southerly movement. We've observed some of that in Texas in the past, where fish have left the estuary and showed up farther south, multiple estuaries following the current over relatively short time frames. And so to what extent does this occur versus fish returning to their natal estuary? How long do fish stay offshore? So the potential for cryptic biomass or an offshore population, and really even in that context, for a lot of the Gulf of Mexico, certainly from Mobile west, I don't want to speak for the South Atlantic, but in the Gulf, nearshore conditions are often very similar to an estuary. And we certainly get southern flounder in the nearshore zone as well year-round. So do fall migrants return to the estuaries the following spring, or do fish spend multiple years offshore before returning to the estuary? So this is a question I've always thought was kind of interesting, because we've had a lot of fish that we've tagged leave and not return. Is that a tagging issue? And what proportion of the fish return to the estuary? We assume it's the same fish coming back the following spring. But what proportion come back versus what proportion potentially stay offshore for longer? There's some questions in terms of how long that might occur, or do fish stay in an offshore population for a longer period of time? And then lastly, the spring migration and the timing and the cues for the spring migration. There's not a whole lot of information on that as well. And the potential for homing to the estuary of origin before the fall migration, or straying into adjacent estuaries. So just to summarize these questions, and this isn't an exhaustive list, but key questions and data gaps that I think exist with southern flounder movement is the timing and drivers of fall migration, location of spawning, migratory contingents, so the skip spawning, freshwater contingents, and the potential for estuarine spawning, offshore movements and behavior, what do the fish do when they're offshore, and then the timing and drivers of the spring migration, including homing versus straying. And then from a management context, I think the big question here then is inter-estuary or interstate connectivity, and what implications that has for stock identification, either at the state level, or do we need management at a broader level, or at least cooperative agreements across states to help address some of these questions. So with that, I'm also the moderator, so I'll stop there.
And you didn't have to cut yourself off.
Well, I thought I might have to. I was getting the signals.
Any questions? We do have time.
Yeah, this is Fred Scharf from North Carolina. So I remember from the genetics work that Anderson Joel did, and it didn't indicate much structure within the Gulf, at least not that I remember. And for a lot of other species in the Gulf, it seems like there's a sort of Eastern Gulf, Western Gulf split in terms of how some of the management takes place that's based on some of the population biology. I just wondered, from the tagging data and telemetry data sort of collectively, is there any hint of that kind of split with flounder?
That's a great question, and we don't have an answer yet. So I know the limited tagging we've done in Louisiana has been west of the river. We do currently have arrays out around the river mouth right now, and so I will be interested to see if we pick up any of the southern miss or South Alabama fish moving around, but we haven't yet. But um, we've talked about that some. So to answer the question in short, I don't think we have that answer yet, but certainly interesting. Yeah. What else?
John Maresca, Alabama Marine Resources. Um, so during the fall migration, these fish are moving offshore as well as most fish coming out of the estuary. Is there any evidence that they may be subject to higher levels of natural mortality, different predators offshore versus inshore? And
That's a fantastic question. We were actually talking about this at lunch yesterday. So I don't think there's, to my knowledge, evidence of that. And if anyone knows of any, please speak up. But anecdotally, I did notice I tagged flounder for a long time in Galveston, and I couldn't help but notice the amount of dolphin activity that would increase in the fall. And we tagged a number of flounder, a large number of large flounder that had dolphin marks, teeth marks on their body. And so we would notice schools of dolphins with their young a lot of times playing with the flounder, throwing them up in the air and letting the younger dolphins chase them. And I think that's what was happening, where we'd get a lot of these fish that were really scarred up. And but the dolphin activity at that time was unbelievable. So this is all anecdotal, but I would not be shocked if there was a heavy natural mortality associated with that. But to my knowledge, no one's really looked at that in detail. I don't know if anyone else in the audience has any information on that, but it'd be good to look at. Yeah.
I wore better shoes today.
Hey, this is Henry Hershey from Auburn. Any idea what plays into a one-year-old fish's decision to overwinter in an estuary versus leave?
No. I suspect that those fish were immature. So when I say one, I mean they're almost one. Probably. But I suspect they're immature. I guess at that size, there is a potential they could be males, but we suspected they were immature females. But no, not sure. At that stage, I would assume that it's immaturity. So.
I think that's it for time.
Okay.
I'm going to ask an administrative question real quick. We're trying to get a head count to give the lunch folks a little bit better idea of how many people will probably stay. I know LDWF suggested some of their folks are going to be leaving even before lunch. Can you raise your hand if you plan to be here for lunch? We'll just do a hand count real quick. Okay. All right. That helps. Thank you.
Cool. All right. For our next talk today, we'll be hearing from Landis Randall at Texas A&M University in Galveston, and she'll be talking about movement and egress patterns of southern flounder from estuaries in Texas.
Good morning, I'm Landis Randall. I will be talking about some of the flounder tagging we've been doing over in Galveston. This work was originally started by Dr. Dance, and Chris Steffen was the master student that started it. Since then, Shane Stevens has also helped out, and I came on last year to continue the project. So there have been quite a few people who's been working on this. So just some quick background. These are estuary-dependent species that adults move offshore. They are highly valued ecologically and economically. They have a susceptible to high fishing pressure, and they have a population declining, as we noticed yesterday. So the objectives of this project were to look at specific migratory pathways, determine which corridors or paths in the Galveston Bay system are used most, to look at the variability and timing of egress and ingress in the system, and see if we can relate those to biological factors, like cold fronts and sea surface temperature changes, and then also to see if the timing of the migration events could influence susceptibility to fishing pressure. So here's our study area for 2016 to 2018. That was when Chrisp started the project, and it was mostly focused on the Galveston Bay complex. So we have in the, just to orientate y'all, because not everyone here is from Galveston. This is Galveston Island. Here is Bolivar Island. This is the Houston Ship Channel. This is the main entrance into the Gulf of Mexico. Down here is San Luis Pass, a second entrance into the Gulf of Mexico. So it's the 7th largest estuary in the United States. It has an average depth of 2 meters. Clearly within the ship channels, it gets deeper. On this map from 2016 to 2018, all of the black dots are acoustic receivers. The blue dots were acoustic releases that we deployed at the jetties of the main ship channel and at the San Luis Pass. So currently, this is what it looks like today. We have expanded the array to increase coverage within the Galveston Bay complex. We have put receivers up in the Anahuac area, East Bay, and down here in West Bay. We've also added 10 coastal receivers, and then we have added 16 receivers up in the Sabine Lake ecosystem. So we've been tagging fish since 2016. There's the numbers: 10 in 2016, 50 in 2017. 2018 was all conventionally tagged fish. 2019, we tagged 35 individuals. 2020 was 50. And then this year, in 2021, we tagged 80 individuals. All of these have been by hook and line using Vemco V9 tags. The delay rate has varied over the years, but it's always been over 365 days. So that we can try to get 2 periods of ingress and egress within the system. And then in 2017 and 2018, we did do some conventional tagging using T-bar Floyd tags. All this are internal tags right in the perineal cavity, suturing them up, giving them some time to recover before releasing them back into the wild. So here is a map of our tagging locations. The green boxes here are the tagging locations. The blue circles are the receiver locations. For the initial 2016 to 2018, we spread out our tagging at known flounder locations. And then since then, the majority of tagging has taken place either at the Galveston Ship Channel, which is down in here. And then all of the tagging done in 2020 and 2021 were at the Tambug campus, which is right there on Pelican Island. So kind of right where all the fish stack up, we can just walk outside with the fishing pole and tag like 20 fish in an hour. So makes it pretty easy. So this detection map is just showing detections from 2016 to 2018. Most of our longest duration to tag detected was 353 days. The majority of our detections were found at tagging locations, which were near receivers, kind of showing limited movement and that individuals remain at the tagging sites for a long period of time. This year, or this is what our detections look like up until, I guess we did our last round of downloads in December of 2021. So as you can see, we have quite a bit of detections in the Galveston Bay. We did not see any Galveston Bay fish detected along the coast or up in Sabine Lake. We do have detections in the Galveston area, up in like the Houston Yacht Club, quite a few around Pelican Island and the main campus, or Tambug campus, and then here at the South Jetty. And we do have some detections down here in West Bay. No detections up in East Bay or Anahuac. So I'm just going to zoom in on the Galveston Bay area. So yeah, again, just pointing out here, the detections down in West Bay tends to be a little saltier than our Anahuac area. Again, trying to explore that connection a little more. And then kind of focusing in on Pelican Island, because a lot of those bubbles overlapped. This dot here is the campus. So you should see we have quite a few in the Galveston Ship Channel, in the main ship channel, on the Texas City Dike, and on the backside of Pelican Island. Here are our detections that we have over the years. Again, 2021, we did a receiver download in December. All of our tagged fish were in November, December of this year. I think we were the only people in Texas that were happy about the flounder closure because we got all 80 tags out before December 15th. So we think that, fingers crossed, eliminated a lot of the fishing mortality that we see some years. All right, so for the 2016-2018 data, we have classified the flounder into migrators and non-migrators. A non-migrator fish is a fish that travels less than 5 kilometers, and a migrator fish is a fish that travels over 5 kilometers. The unclassified fish are fish that were detected at a receiver and then never detected at any other receiver. So as you can see, the majority of our fish, of our flounder tagged, are unclassified, but we do have 10 that are non-migrators and 15 that are migrators. We do want to continue this for the following years, and that is a work in progress. So here's some examples of what the migrators look like. In figure A here, oh, man, this is hard to use, sorry. In figure A, the fish was tagged off of Texas City and then migrated out through the Houston Ship Channel. And in figures B and C and D, the fish were all tagged on Tama campus and then migrated down the Galveston Ship Channel and then out through the main jetty to the Gulf of Mexico. So as we saw in this data, that the movement times were very rapid and quick. So within 9 to 27 days, these fish were tagged, picked up at a receiver, and then picked up at our south jetty receiver. So that's a relatively quick movement in a short time frame. Here's some examples of non-migrators using the 2016-2018 data. The non-migrators account for about 78% of our total detections, so they're the majority of what we see. Kind of all these were tagged on the Tama campus and just kind of migrated, moved to and from this little area, maybe the backside of Pelican Island. They just kind of hung out in this area. And like Mike said in his talk, some of these fish we thought were dead, but they weren't because we were still catching them. So we did conventionally tag some fish in 2016 or 2018 and 2017. We had about a 12% recapture rate for 2017 and a 13% capture rate for 2018. This one was cool. We did conventionally tagged a fish in Galveston, and then a fisherman picked it up south of Port O'Connor. About 23 days later, which is, we estimated a distance of 231 kilometers straight line distance, like we measured the arrows on that figure. So that means that fish was traveling about 10 kilometers per day, which is, I don't know, flounder don't move very fast in my opinion, so I thought that was pretty wild. We have been looking at the timing of egress, mostly just focusing on the 2016-2018 data. So we are seeing the Y column, the Y-axis is the proportion of migrators, so it's just looking at those 10 fish that migrated. So that's the proportion of migrators that were moving throughout the year, and as you can see on this graph, the majority of them are moving in November and December. We're not seeing a lot of movement in the spring and summer months. So we have found evidence of different migratory patterns. Some are residents and some are displaying migratory behaviors. We are seeing that egress begins in November and continues through December into January. This could potentially lead to a mismatch in fishing regulations and fishing pressure. And we are seeing some evidence for interbay connectivity with the one fish that we picked up down in Matagorda, but we're hoping with the expansion into the Sabine and the coastal receivers that we can start figuring out what's going on a little better. So for future directions, we want to further investigate the effects of environmental parameters on the movement and timing of flounder migrations. So for the 2016-2018 data, we did see that as seawater temperatures declined, we saw an increase in the average distance moved. So kind of continue this for the up until the current year that we've tagged, see if this pattern holds true. We also want to look at the spatial extent after egress. Where do they go? Do they go north? Do they go south? And then look at the effects of ontogeny on movement. For the 2016-2018 data, we divided them out into size classes between 40 and 49 centimeters, and then 50 to 60 centimeters. And we saw that there was, that the smaller fish remained here. The smaller fish remained in the estuary significantly longer than the larger fish. So we want to see if this pattern holds true up until 2021. And then we do want to determine the potential for egress. So far, we have not seen any of our tagged fish come back into the Galveston Bay complex. We see them leave, and we don't really ever hear from them again. So hopefully, maybe this year we got a little bit more tags out than we ever have, and there was a flounder closure, so I'm optimistic. I think that's sometimes all you need, is just one person being optimistic. Again, just want to thank everyone that's helped out with this project. Chris, who started it, Shane, who's our fishing guru and has caught most of the fish for me this year and in previous years. Tom and Mariah for maintaining the receiver array. Jansen for helping tag fish, and then previous grad students in the Rucker Lab. And with that, I'll take any questions.
Hi, Russell Vorsky from North Carolina State. I'm curious, do you have any data on sex differences in
I
this
do not.
movement? Okay.
We are tagging, I don't think I mentioned, we're tagging fish over 17 inches, so we're assuming they're all migratory or migrating females.
Okay. Okay, thanks.
Yeah.
Nancy Brown Peterson, Southern Miss. That might have just answered my questions, because I note that you said that the smaller fish stayed in the estuaries longer, but I was wondering if there was an actual difference in the size of the fish that did migrate. Is there a significant difference? Is it larger fish that are migrating out?
Okay.
Smaller fish maybe aren't migrating as much.
I don't think we've completely teased that one apart.
Thanks. Sean Powers, University of South Alabama. The Tamug campus, are those fish, do you think, resident there, or are they already staging to move offshore? Because I spent some time there. I've been chased away last time I tried to fish there by the campus police. But it's true. Are they staging there, you think, or because it's so close to the inlet?
I think they're staging. We did have a couple tags returned to us this year from fishermen that killed flounder after the closure was ended. And I went back out to see if we could put these 2-3 tags in, and within half a day, and we only caught one. It was not the fishing rodeo that it is during the run.
So I guess my question though is, where are those fish coming from to Galveston? Are they coming from the upper bay, or y'all don't have enough detections yet?
We don't have enough detections yet. But next time you come to campus, if you want to fish, just we'll tell campus police to leave you alone.
I tried to use Mike Dance's name, and they just really kicked me off.
Coming, Eric.
Have y'all thought about spreading out your tagging locations, initial spots that you tag, so that I noticed that you had a lot of, and Mike hit on this, a lot of tight residency around one receiver.
We have thought about spreading out tagging. Initially in the 2016, 2017, 2018 years we did, or 16 and 17, we did spread out our tagging locations. I was told to focus on the ship channel this year because we really are hoping to try to get fish that are leaving, because we want to see the timing of leaving and hopefully seeing those fish come back in.
Right.
So that's what I was told to focus on this year. But I know it
I
is
think if
a.
you put some fight nets around San Luis Pass, you might load up. So
Sorry, what was that?
if you put some fight nets around San Luis Pass, you
Oh
might
yeah.
load up. Just saying.
We definitely looked into purchasing some pike nets. Okay.
Great presentation, Mike Leffler, North Carolina. Just a quick question. Have you thought about attaching an external tag to each one of these fish that you acoustically tag? That way, if you see these fish emigrate offshore and come into another estuary, that you might get an angular report of that tag instead of an acoustic detection.
They are all acoustically and externally tagged. I skipped that part.
What
But we
type
do
of
have like a Floy tag with our contact information on it. So if they do catch it, they can call us.
what type of Floy tag are you using?
A T-bar tag?
Okay, thank you.
Oh, no? Okay.
I think that's it.
Okay.
Thank you, Landis. For our next talk today, we'll hear from Jennifer Green, who will be talking about migratory movements of southern flounder in Mississippi coastal waters.
Thank you. Okay, try this. Good morning. I'm Jennifer, and I'll be talking to you about migratory movements of southern flounder in Mississippi coastal waters. This is a collaborative project between the Mississippi Department of Marine Resources and the University of Southern Mississippi. We just got started last year, and so this presentation is just going to be a preliminary look at some of the initial detections that we have so far. And so southern flounder support valuable recreational commercial fisheries throughout their range. Throughout recent years, we've seen declines in abundance and harvest between recreational and commercial fisheries. And for Mississippi alone, you know, we have the same pattern of this trending decline. So these figures, these figures just represent the recreational and commercial harvest of southern flounder in Mississippi from 2012 to 2021. And although there is annual variation, overall there is a trending decline. And so similar patterns have been observed in fishery independent trawl and gillnet surveys across multiple states throughout their range. And this is increased stockholder concern about the status of the southern flounder stock, which has prompted several states to make regulation changes to size limits and bag limits. And even some states such as Texas, Alabama, and Florida have even implemented seasons of no harvest, which typically coincide with their spawning period. And while we do know that they migrate offshore and spawn during the fall, detailed descriptions of their spawning migrations are lacking. And so prompted by these range-wide evidence of decline, MDMR recognized the need to develop projects to increase the knowledge of the stock. And in 2018, DMR implemented the FICENet survey to identify or to look at southern flounder abundance in Mississippi, which Josh talked about that a little bit yesterday. And then last year, we began this collaborative telemetry project aimed at documenting the timing of these movements and environmental drivers behind southern flounder movements. Ultimately, for this project, we would like to accomplish we would like to figure out the timing of southern flounder migration, so the emigration from the estuaries to offshore spawning areas, and so figure out when that timing is for the three major drainages in Mississippi, which are St. Louis Bay, Biloxi Bay, and Pascagoula Bay. We'd also like to determine the timing of when these individuals are returning to the estuaries and look at the occurrence of possible estuarine homing. So are they returning to the same estuaries in which we tagged them, or are they going elsewhere? And lastly, we'd also like to evaluate potential environmental factors influencing southern flounder movements. As I mentioned, we just got started last year with this project, and so this is a current and ongoing project. And so for the purposes of today, for the purposes of this talk, we wanted to, our objectives are to use the first 6 months of our detection data to provide a summary of the first year of tagging activities, to describe broad movement activities of flounder within the Mississippi Sound, and to determine the occurrence of these immigration movements to potentially offshore spawning locations. Sampling occurred from early August to the first week of November in 2021 using pike nets and hook and line sampling. And we were targeting flounder that were at least 12 inches in total length. Selected individuals were externally tagged with an acoustic transmitter and a dart tag. The transmitters, they were a V9 transmitter and battery life of 104 days. And so, again, like the previous talk, we're hoping to get 2 of those migration patterns, the egress and then the ingress. As with any telemetry project, acoustic receivers were deployed to detect the transmitters. And this being a collaborative project, we really took advantage of the extensive acoustic arrays that were already established throughout Mississippi Sound. These are managed by several agencies, which include the Gulf States Marine Fisheries Commission, Mississippi Department of Marine Resources, USM, their Center for Fisheries Research and Development, and the USM Estuarine and Movement Ecology Lab. And for the purposes of this talk, we grouped receivers into 4 zones based on geographic area, and I'll be referring to these zones throughout the rest of the talk. And so the west zone includes those receivers around the St. Louis Bay area. The central zone included receivers from the Biloxi Bay area. The east zone were those receivers in the Pascagoula area, and the outer sound were those receivers that were furthest south and around the barrier islands. And it's also important to mention that all sampling and tagging efforts occurred within the inshore areas within those west, central, and east zones. And here's just a quick tagging summary. So from early August to the first week of November, we tagged 96 individuals, and we can see summary of that in this top figure here, where it just shows the number of tagged individual flounder on the Y and month across the X. And you'll notice that most of the individuals that were tagged occurred in September and October. We tagged 36 individuals in the west, 43 individuals in the east, 43 individuals in the central zone, and 17 individuals in the east. I also have here that sex is currently undetermined. While we were tagging, we did retain fin clip samples, just in case we're able to eventually one day potentially use genetic analysis to determine the sex of these individuals. And the bottom histogram shows the length distribution of the individuals that were tagged. The size ranged from 301 to 500 millimeters in total length. And again, we have the number of flounder tagged on the Y and the size bins across the X. You can see it's a fairly normal distribution. And this dashed line represents the length at 50% maturity as reported by Corey et al. In 2017. We included that there just to show that most of the individuals, or nearly all the individuals that we tagged, were likely mature and therefore more had a higher potential of making those offshore movements. The environmental conditions during tagging, the temperature ranged from 18.7 degrees to 33.7 degrees Celsius, and we had a fairly broad salinity range as well, ranging from 0.2 up to 22.5 ppt. So during the first 6 months of our study, from August through the end of January, we've recorded well over 200,000 detections from 84 individuals, and so we've heard back from 87.5 of the flounder that we tagged. And the abacus plot on the right represents the daily detections of individuals detected within our array or within our study area. And so here we have transmitter ID on the Y, which you're not really going to be able to see. It's okay, that's not very important for this anyways, but just know that each row is an individual that was detected, and we have date going across the X down here, and the points represent each day that individual was present or detected within the array. And so you can start to notice variability between detection periods, where some individuals are detected for a few days, and then they leave, we don't hear from them, and then we hear from them again months later or weeks later, whereas other individuals are consistently detected for consecutive months at a time, which we've heard about that already this morning. So we are currently in the process of going through and identifying the individuals that are detected on multiple receivers versus those that are detected on consecutively on single receivers. Initially, we thought to weed out potential mortality or tag loss. However, after listening to the talks this morning, that might not be the case. Then we also wanted to look at transitions between those inshore zones and to identify if fish from Pascagoula were being detected over in the west and vice versa. And so again, we have abacus plots to the right, set up in the same way with transmitter ID across the Y, date across the X. And this time we've grouped them based on where individuals were tagged. And the color represents where the zone in which individuals were detected. And so it looks like there's a lot going on, but focusing on just the color, a single color means that that individual was detected in one zone. Multiple colors means that the individual was detected in different zones. So looking at the west, looking at the top here, these are individuals that were tagged and released in that west zone. And you can see most of them, pretty much all of them except for one, were detected in the west. Jumping down to the bottom abacus plot in the east, those individuals that were tagged in the east primarily remained detected in the east, over in the Pascagoula area, except for one individual that transitioned into the Biloxi area. And then in the central area, the majority of individuals stayed within that Biloxi area while they were inshore. However, there were a few individuals that transitioned into the east and remained in the east, or moved back and forth between Biloxi and Pascagoula. In general, what we've seen so far is limited mixing between those, between the major estuaries. Now looking at the seasonal occurrence of where these fish were detected throughout the first 6 months. These bubble plots represent the number of individual flounder detected during each month, and we noticed that there was an increase in the fish detected in the outer sound during the fall. Again, and that's so there are a few individuals detected during September and October. However, by November, the number of individuals detected out around the barrier islands and in these passes drastically increases, and that carries over into December, which seems to coincide with what others have presented this morning, and then drops off again in January. And I included the table there just as a reference to remind folks that from August through November, we were adding tags into the system, and the number of individuals that were detected. And so real quick, there was 39 individuals detected around the Barrier Islands. Most of these movements did occur in November. And in this figure, we have the last inshore detection and the first detection around the Barrier Islands, and the lines just represent estimated travel corridors or movement pathways between the inshore and outer sound areas. And for the most part, they seem like they're pretty directed movements in the sense that flounder are usually detected in the passes that are adjacent to where they were tagged. And so just to recap, we heard from just over 87% of the tagged individuals. There was limited transitions between those inshore zones, and the majority of, or higher numbers of individuals were detected out around the Barrier Islands during the fall, and that coincides with what Morgan et al. Found with elevated gonadosomatic indices. So this was pulled from Morgan et al. This is the GSI, or the gonadosomatic index for sexually mature female flounder from north central Gulf of Mexico, and you can see those values start to increase in October, ramp up or peak in November, and then start to decline in January. And so our findings so far seem to kind of follow that pattern as well in terms of when these fish are moving out. We're continuing this project, and this year, again, we want to look at those environmental drivers. We do have data loggers deployed to try and look at potential cues for temperature and salinity. And I think I'm pretty close on time here, so just to give acknowledgments to our funding sources and collaborators, without everybody, we wouldn't be able to conduct this research. So thank you.
Questions? See Mike and Nancy. Can I start with Nancy? She's my academic mama.
Yeah, that's true. Nice job, Jen. This is Nancy Brown Peterson, Southern Miss. So in January, it looked like you still had some fish in Bay St. Louis. Are they smaller?
We'll have to look into that. At this time, I didn't look yet. This is all preliminary. So that's certainly a question that we are looking into. I will say that some of those early detections around the Barrier Islands in October, they were actually from those smaller sizes that we tagged. So.
Hey, great talk. There's such an interesting dynamic to me in Mississippi, in that you have the estuaries, but then you also have the barrier islands. So, and you may not have the answer to this yet, but do you think those fish, just based on the plots I saw the other day, are actually moving through the passes from the barrier islands and farther offshore? And I guess what constitutes kind of the estuary, do you think, for those fish? Where's the barrier, I guess?
Right. And again, that is something that we've questioned as well and are wondering. It seems like they are stacking up in those passes. However, looking more closely at the detections, there are several individuals that just kind of circle around the barrier islands during November and December. And so that might be, their where it ends for them. We're not sure. It's still early. We're hopefully be able to get more information in the spring when we do our next download. So it'll be interesting to see if there's data gaps, how long individuals were gone, if we hear back from them, when that timing is, and if they're continuing to come through the pass or just kind of hang out around the north or south side of the islands. I don't really think that answers your question, but it's all very interesting, and we're excited to see what we get this spring.
Okay, thank you.
Thank you.
Thank you, Jennifer. For our next talk, we'll hear from Dylan Kiene of South Alabama, and he'll talk about describing southern flounder spawning migration from Mobile Bay via ultrasonic telemetry, timing, rates, routes, and homing.
Walk around a little bit, probably. All right, so I'm Dylan Kiene. I'm a PhD student at the University of South Alabama, and I'm going to be presenting some work that we did with acoustically tagging flounder in mainly Mobile Bay, but a little bit in Mississippi Sound. I'm going to try to cover a lot of information. So I think this is still not the one that I uploaded this morning. Okay. Sorry, I made a few last minute changes. So I'm going to try to cover a lot of information and then leave some time for some questions. So we're going to go over years 2019 and 2020. I'll touch on the design a little bit, give a summary of the overall detections and how these fish are utilizing latitude during this time. Look at some of the rates at which these fish are migrating and not migrating, and some of the routes that they may be taking. And then look at a comparison of the size in the non-migratory versus non-migratory flounders, and then talk about this homing characteristic that we observed a little bit of. And so this is the receiver array design that we went with starting in 2019. We're essentially encapsulated Mobile Bay in such a way that the fish could not leave without being detected. So we've got the delta up there covered, all those passes. These tidal rivers, which we know are really, really important for flounder during this time, mainly Dog River there is where we tagged a lot of our fish. And I underestimated the importance of the Mobile River during this time. I didn't realize how many fish we'd end up tagging in there, and it's a difficult place to actually put receivers. But we do have some in there now. Then the industrial canal here, and then this is Fowl River here, which is a more natural river system that has an opening here at East Fowl River in the bay, and an opening here, what they call West Fowl River, that goes into the Mississippi Sound. And I'll talk a little bit about the fish that we did tag in this system, and then a gate at the Dauphin Island Bridge, and then at the mouth of the bay. And so we went with a Vemco V8 acoustic tag on high power with a ping interval of random interval between 60 and 120 seconds, which is pretty fast. But I didn't want any chance of these fish being able to swim through that gate without being detected. And we are surgically implanting these fish from the top or the brown side, basically at the base of the gonadal cavity. So I can actually, there's a sweet spot there that the tag lays in very nicely, and you can actually look at the sex of the fish this way too. And these are all females from what I can tell macroscopically. And there's just a little double overhand stitch there. Flounder are the best surgical patients I've ever worked with in terms of fish. They're very nice and calm. And so this is the size distribution. You'll see that gold dashed line on all these figures. That's just the new legal size, which is 14 inches. These are all half-inch size bins. So we got a few undersized fish. These just get a mark recapture tag, and then all these individuals are carrying acoustic tags, which for 2019 was 55 individuals, 50 of which were in Mobile Bay. You'll see over here mainly in Dog River and the Mobile River, a little bit in the Industrial Canal, and then we've got another 5 in West Fowl River. And one of those included this nearly 10-pound. This is Captain Richard Rutland, the guide that we used for this. I should mention that this is 20 days worth of fishing effort on an 8-hour charter. So you can see that these are some expensive flounder for 2019. And I love the way they swim away at the end. It's really nice when you see that. And so this, I could probably spend the rest of the talk just talking about this plot. But this is all the detections over time with monthly time steps on the x-axis and all the individuals on the y-axis, and then the color scale reflects latitude. So the dark purple detections, which hopefully you can see from out there, are detections that are on all the receivers that are in those tidal rivers in the northern part of the bay, and those orange detections are detections from the receivers at the mouth of the bay. It's about from Dog River about 30 miles, from the Mobo River about 35. And we observed 15 of the 50 flounder to be migratory out of the bay. The average size for this year was about 17.7 inches. But these flounder did range in size from legal, right at 14, up to 22 and a half. You'll notice you could probably start to see some interesting things going on here. So we got egress from pretty much 100% egress from the rivers during the early parts of November and some a little bit in October. But then not all of these fish are actually migrating offshore, which was mind-blowing when we got this all back. For me, at least, I would have expected that all of these fish would. It was kind of a cold winter for us a little bit too. And then you see actually we did pick up, the study was not really designed to look at it, but we did get some returning fish. And in fact, these fish are returning to the exact same river systems in which they were tagged, which was super, super interesting to me. Like this fish left Dog River in November, returned 6 months later. Here we'll look at the Foul River fish in a minute, but one of those did that as well. And then we've got some offshore migrants that actually returned. And the rate varies at which they, from the timing of egress to the rivers to the passing through the gate. The fastest fish to do it was the 6-pound flounder. Did the 30-mile swim in 3 days, and then actually came back to us in April and started working her way back up the bay, which was really interesting. And then we have this poor fish right here is an interesting story. It is one of the, sadly, it was probably confused. It didn't leave till January, and then kind of came back a month later, realized it was maybe a little early, left again, and came back. And then the night it came back, it got gigged. And so we have that. And another way to look at this would be to look at mean latitude usage by these fish, monthly mean latitude usage by each individual, and just note that this jumps from month 6 to month 10. So if you start in October, you see all these fish using these higher latitudes. And then as you get into November, you start seeing them come down and into December. And then as the new year progresses, we get some coming back. And this is the West Fowl, and this is it's interesting to me because the East Fowl and West Fowl basically operate as independent systems. There's no connectivity for these flounder. They don't swim through the entire river. And then you can see egress occurred from the river throughout November, and then we did get one fish returning to Foul River in 6 months later in May. And we did not observe any eastward movement from these fish. So that if they did migrate, they did not go this way. They would have had to go out into the sound where we did not have great coverage. And so this is 2020. We did we actually captured we tagged 70 fish this year, 45 of which were acoustics. We got a lot of smaller, undersized fish, which we were pretty excited about actually. Maybe seeing some indication that there was successful spawning with the first year of November closure. And then we do have some size gaps in here, and then a lot of larger fish, which is good. Similar tagging locations, the Mobo River, Dog River, some in East Fowl, and then some in West Fowl. And unfortunately, this plot is not quite as pretty for this year due to the loss of a receiver at the mouth of Dog River that kind of smooths out that exact egress from the rivers. But the story is generally the same, except now we see some overwintering in the tidal rivers by some of these individuals, which is really neat. And they're actually occupying spaces further up into the river. So if you saw in the last plot, there was a lot of detections periodically, and those fish were actually using about the first 1,000 meters of the river going up and down. And I may try to look at tidal phases over that time to see if they're moving up and down with the tide. But yeah, and then we see some more homing activity where fish leaving individual river systems and then coming back to those exact same river systems, which is interesting. And then we saw, these are the West Fowl detections I'll blow up in a minute. and then some migratory proportion of these fish. And this is a conservative number. I expect this to come up a little bit. We've got 2 receivers that are attached to structure in the mouth of the bay, and I just haven't been able to get divers out just yet and get those back, but I will expect, we've seen detections on those receivers before. Oop. There we go. And this is mean latitude again by month, and it looks a little bit different this time because of the overwintering fish, both in Fowl River and in those northern tidal rivers. But you do see some progression, and then the tag, we deployed a lot of these tags a little bit earlier, so we kind of probably didn't get that ingress activity that we did see in the first year. This is the West Fowl River detections again. Most of the fish leaving around late October to early to mid-November, and then we did have one overwinter resident that stayed all winter long. So pretty neat. And this is probably what most people are interested in, and this is the detections through the gate at the mouths of the bay. And so you can see that most of these fish are leaving during November, but some do a little bit early, October, and then December, and then a little bit in January. And you do see a little bit of return action by this individual, and this individual, which is pretty neat. And so these are some of the routes. R doesn't seem to recognize Gilliard Island or the land over here as problems for flounder, but the point of this is basically that I would have expected these fish to either utilize the ship channel or either shoreline as they went on their way out, but there's no real clear indication of that. They just are heading out, and we basically got detections on all the receivers. And if you actually look at this seasonally, you can get a better idea of what's actually going on. Most of these fish are utilizing these paths before the end of the year, and except for the one that came out of the Mobile River in January and came back over here where he was gigged. But there does appear to be some indication that they orient to the Dauphin Island Bridge when they come back in the new year, before going back up into some of the areas that they like to be in the summertime. This is the same thing for 2020, and this is biased to the way that the receiver array is designed. We didn't have a whole lot of coverage on the eastern areas of the bay during this time. But again, there was maybe a preference to the eastern side there for this year. We did get one that left, out of the Dauphin Island Bridge. But all these fish actually did so before the end of the year. And so this, Nancy, I'm sure you were interested in this. I was too. And so this is one way of looking at the size on the x-axis of the purple, which is non-migratory flounder, and then the orange, which would be migratory flounder. And there's really there may be some pattern here where these would be the first time, about the size at which they would first be maturing, maybe the most gung-ho about going out. But another way to look at this is with box plots, and actually there's no statistical significance between the difference in size between between resident and non-resident flounder. The migratory portion was a little bit bigger in 2020, but not statistically significant. This is the one recapture of the sad fish where he got gigged the night he came back. Might be a prime candidate for microchemistry though, so kind of hold on to him. But these are some. We had 10 of the 70 flounder that we tagged in 2020 recaptured, so they do have mark recapture tags on them as well. And what I want to point out is that there may be some, this kind of validates the fact that the homing and then the residents in certain rivers that they're like, but there is some benefit in growth potentially for fish that do decide to overwinter. So you see some of those growth rates. Hopefully it's clear up there, but the coolest one to me is this fish here, 13.5 inches in October, 220 days later at 20 inches, and that's real. I got hands on that fish. And all these fish are recaptured in the same exact system. Mainly Dog River seems to have, these fish seem to be having the most success with growing. And these are overwinter growth rates, which is really cool. And it's also the time at which these fish might be maturing and able to allocate resources to that. And again, 324 days, they're about 5 inches recaptured in the same river systems. All these fish were basically recaptured very close to each where we tagged them. So kind of in summary, we observed migration rates of 20 to 30%. I do believe that might be conservative, at least for the time being. The timing of egress, the majority of these fish do leave during November, but there is a few that go out in October, December, and January. No statistically significant size difference between migratory and non-migratory flounder. And in terms of routes, 90% appear to egress through the mouth of the bay and only 10% through the bridge and into the Mississippi Sound. And there were no observed from west to east movements from the flounder from West Fowl River. And so I think this study really wasn't designed to look at this, and I was really surprised to see this. It was kind of right as these tags were dying that we were picking this up, but this homing appears to be potentially really, really important, and maybe it has implications for how we manage these fish, and also for hatchery practices. You know, where you let your fish go might actually be really important if there's any sort of imprinting or orientation back to a certain area, and really highlights a need to identify offshore spawning grounds in the Gulf and in the Atlantic as well, and then figure out what role physical mechanisms are playing and where, you know, if the adults are orienting back to the areas that they already occupy, where the juveniles and the ones that are being spawned coming in. And so the other questions I have are, you know, what's going on with the large number of fish not migrating? Is it some sort of population contingency where we see maybe a freshwater resident contingent, offshore resident contingent, and some sort of migratory in between contingent? Is there high mortality associated with the spawning migration? This could be a thing, but we know it's likely it shouldn't be fishing related due to all the closures. And during this study, it was closed in Alabama. Fishing was during the month of November. But it is something I've considered. And then skipped spawning. This write-out paper is pretty neat. You know, I also read a scary paper one time about how acoustic tags can act as dinner bells for bottlenose dolphins. And so that was something I think about every day pretty much. And then, you know, is there any kind of spawning adaptation? I think we should ask that question too. You know, are eggs being released in the bay, or are there suitable habitats now with deep ship channels and or nearshore structures? We've got the Alabama Artificial Reef Permit Zone, which provides a lot of nearshore structure and also could potentially bring predators into the area that they may not want to fight with, you know, and then it kind of all ties back into the contingency stuff. But we kind of hope to answer some of these questions with the 2021 update. We went to a V9, which has a full year worth of tag life, and redesigned the receiver array a little bit, pulled those ones out of Fowl River where we knew there wasn't any connectivity, moved some into the bay, put one over here, put some over here where I think is important, and then I should have extended this. We actually put some over on the west end of Dolphin Island on those CCA reefs, and then one over here in the middle of the sound. But we had a really successful year with the tagging. We actually captured 300 flounder in the 20 trips, tagged 255. A lot of really small individuals, which I'm curious about. We did give a number of these to the hatchery, and they determined they were pretty much all male at that time, which is interesting. But we also saw a lot of really, really big flounder, and we did tag some really big individuals, kind of all over the place. And all the acoustic tags, though, were actually in the bay. I think that's it, but I might be frozen again. Just want to acknowledge some folks real quick, hopefully. Yeah, I really want to acknowledge my advisor, Dr. Sean Powers, for all the support he's given me throughout this. Captain Richard Rutland is an incredible fisherman and been really, really instrumental in this project, getting all these tagged fish. Our lab manager, Crystal Hightower, and the entire Fisheries Ecology Lab. You know, for that design and the receiver array, we were using divers to put all those receivers out on structure, and she is one of those divers and really instrumental in that. And then Alabama Marine Resources Division, John Maresca and Kevin Anson are here. Really appreciate you guys. And then National Fish and Wildlife, and then Alabama Pipe and Supply for donating the concrete moorings that we use to more the ARs. So I forgot to mention that we use acoustic release receivers to make that gate using a mooring design that we've had success with offshore. And so that's it, and I'll take any questions.
John Godwin, NC State. Really nice talk. And it's interesting with the homing, and maybe I missed it. Did you see any fish moving between? Between going somewhere else, like another of your
a
locations?
little bit, but they did. They at least this was this tags were dying right at the time that this was happening, unfortunately. But we did see that 6-pounder, for example, when it did come back. It kind of went up the Dolphin Island Bridge and then was detected at the mouth of the East Fowl River and then eventually at Dog River. So it did eventually go back to its original tagging location. But they probably occupy a lot of these tidal river mouths. They're just really suitable habitat for the way that they like to feed and stuff, especially in the fall. That's a good question, though.
Hey, Dylan, nice talk. Troy Farmer from Clemson. I was just curious if there was any spatial component in your tagging with the probability of fish migrating. In other words, did fish that you tagged up in the Mobile Bay and Delta, or Mobile River and Delta, were they less likely to migrate offshore and fish in the lower estuary more likely to migrate?
It did seem that the most likely migratory fish were from that Mobile River and Dog River, but it's a little bit more difficult to tell. We didn't tag a whole lot of fish in that lower part of the bay during these 2 years. We did in 2021 because we found so many fish everywhere. But during those years, flounder were kind of a tough thing to come by, especially with how many we wanted to tag. And so we really spent a lot of time in those 2 rivers, which proved to be really, really important for the bigger flounder that we were after. But it's a great question. Something I would like to look at.
Hey, great talk, Dylan. I had a couple kind of related questions. The first was real quick. Just what % of the fish that left returned? And I know you had some of the tags were dying, but
yeah, I need to look at that. And it's hard to say for sure because I don't know exactly if we missed some, which we probably did because of the life of the tag. But I think we got 4, maybe 5 of the 22 migratory individuals that returned. So a decent percentage in terms of that's the question I hope to answer this year with a full 1-year tag. Something I'm really interested in.
And the other question I had was kind of likened to that, but you've got kind of interesting dynamic where you have fish in these river systems. And it looked like you have some fish that migrated from the river, but you didn't necessarily detect them at the mouths.
Right.
Any idea where those fish are going?
Yeah, that's the biggest question I have, and makes me. That's why we put receivers into the middle of the bay this year. But we did not have receivers in the Mobile River, which is the deepest river down to 100 foot in some areas in there. Could offer somewhat of a thermal refuge for this fish. I work with a lot of fishermen that do catch big flounder every now and then in the wintertime, a lot of which are from that Mobile River. But there's also areas within Mobile Bay that could, are deep enough to, like the inshore rigs. There's some natural gas platforms out there, and then the ship channel is really, really deep. And so they could be occupying some space in there because we did have a couple individuals that migrated out of the river but not out of the bay, but then would come back to the river 6 months later. Right. So they didn't die. They didn't migrate out, at least as far as we know. And so, yeah, big mystery.
Hi, really nice job. This is Nancy from Southern Miss. So I have 2 completely unrelated questions. You said that because you're embedding them, you can actually see the gonads.
Yeah.
What time of the year, were you doing them like September, October when they might be starting to come up? I'm just wondering if some of the ones that you find staying that you saw that they actually had developing gonads.
That's a great question, and so I'm doing a maturity and an aging growth on top of this to hopefully maybe look at the migration in terms of that. I haven't actually processed any of the histological samples yet, so I'll probably be reaching out to you when I do because I think it'd be really cool if we could standardize that and look at it across the whole northern Gulf. But no, I do a lot of a collection from flounder, especially during October and November, and yeah, there's a significant amount of development in a lot of these fish, and you can usually tell just by looking at them if that area is swollen. But during October, it's not as prominent. until you start getting really towards the end of the month. It happens pretty quickly.
Yeah.
And some early November individuals.
Great. And then just sort of a follow-up, you mentioned the artificial reef zone. Zettelmeyer's got tons of receivers out there. Has he picked up any of your fish?
You know, I haven't reached out to him. That's a great option. I need to do that. And we will have some offshore receivers, I think, this year with a red drum study that we've got going. So hopefully we pick some up.
Yeah, I think you answered my question. Phil Stevens from Florida. Yeah, I was struck by the number of fish that did not get picked up at the gates, but were absent. And I think you touched on it.
Large fish too. I mean, you could see that some 20 inchers that did not.
Yeah, and you look like you had pretty good coverage of those passes. So yeah, I was just wondering, anecdotally, if people are seeing, them aggregate in places that are not the pass, and how much inshore spawning could be going on. And I think you already touched on that with some of the deep channels.
I'd like to put a little bit of effort into actually figuring out. Mobile Bay is really big, and it'd be tough to narrow down exactly without, by trying to fish for them or something. In the wintertime is really not great conditions most of the time in the winter to be out in the middle of the bay. But we're hoping to pick those up on receivers that we deployed this year.
Yeah, I mean, you seem pretty dialed into the angling community, so it seems like they would have some idea of maybe where those spots could be.
Yeah, I manage an angle-based tagging program, Tag Alabama, so that helps a little bit, being able to reach out to those guys.
We're right up on a break, but I just have one quick question. Because I'm doing triple tail and my tags all died, do you get many returns after the tag dies? In a mortality or a fisherman catch?
A few, but not very many, no. And I wonder about that because, we didn't have a lot of recaptures in 2019. The sentiment around the fishery really wasn't great either. People weren't spending a lot of time fishing for flounder, but then as soon as they started to think it was a little bit better, we started seeing a lot of recaptures. But I still haven't got recaptures from a lot of those fish, and I'm curious about that, whether it's high mortality or the tag shedding. I do know that we're using this T-bar tag, and the reason I went with that was to try to not affect the fish's swimming ability at all with this. If I was really running a tagging study where I wanted to look at some of that, they have those loop tags, I think, that are pretty much 100% retention. I think that would be neat. Do some high dollar work maybe with that. Try to figure that out. And who knows how many of these don't get reported by people who take them in the middle of the night or whatever.
Good. Thank you.
Thanks.
Thank you, Dylan. We have our break now, so we have about a 20-minute break. And I guess, Steve, we're at 10:30 now. Do you want to come back at 10:50 or?
Well, because of the YouTube, I just as soon actually make it about a 10-minute break. Come back at 10:40 to try to get back on schedule.
Okay.
And then if I could I have Sydney, Jared, and Patrick come out to their posters? I never did make it out there and get your photo by them last night. Thank you.
Okay, if everybody could begin finding their seats, we'll go ahead and begin the next session, or part of this session. Our next speaker will be Jared Chrisp. He's going to talk to us about exploring Southern Flounder contingent diversity, contribution to fisheries, and correlations with growth and condition using natural tracers of habitat use.
So the talk I'm going to be presenting today is my master's research from Clemson University, where we were looking at the different contingent types across Alabama's coastal waters. So as we all know, Southern Flounder are estuary-independent species, but their actual utilization within these habitats seems to be highly variable across space and time. Over the last several years, there's been several talks about different contingents or different utilizations of salinities within estuaries. A lot of this comes from the incorporation of otolith chemistry into these recent studies. However, otolith chemistry has several limitations. You're able to differentiate between freshwater and about 1 part per thousand, but once you get above 1 part per thousand, you can't really delineate anything across the entire salinity realm that we usually see within estuaries. So we decided to include otolith chemistry and stable isotopes to see if we could tease out any of these differences that these contingents may be exhibiting across Alabama. So we broke our study, or our species, into 3 different contingents. The first one is a transient individual. So we're considering these individuals that are the larval ingress into freshwater habitats before beginning their growth and development within these freshwater habitats, then moving to estuarine habitats to finish their juvenile growth phase before moving offshore. Next, we have an estuarine contingent, and these are individuals that never utilize freshwater at all, but spend their entire juvenile growth phase within estuarine habitats. And lastly, we have the freshwater contingent, and these are individuals that only use those estuarine habitats as a corridor between freshwater and offshore spawning habitats. We had 4 main objectives with our study. The first one is just to understand the proportion of contingents that we're seeing within Alabama's coastal water for that early settlement age, life history age. Next, we wanted to see if there was any effect of river discharge and see if we can predict the proportion of contingents based off the overall annual discharge. Then we wanted to look and see which of these, or what proportion of these contingent patterns are contributing most to the commercial and recreational fishery. And then lastly, we wanted to see if there was any sort of growth incentive to be categorized into one of these contingent types. So our study area is the Mobile-Tensaw River Delta and Mobile Bay within Alabama's coastal waters. It's a unique habitat in that it has a very strong salinity gradient from north to south. So for that, we broke our study into 3 different regions. The first being the Delta, which is freshwater year-round. The next is Upper Bay, which is that transition in between that freshwater and estuarine habitats. And lastly, Lower Bay, which sees that salinity above 3 parts per thousand throughout an annual cycle. So here's more on that salinity. As you can see over the years of our study, that Lower Bay never experienced salinity less than 3 parts per thousand. That Upper Bay seems to see a high salinity peak in the fall. And then, although we have small samples, we do showed that our delta was freshwater year-round. So our sample collections come from 04 to 07 and 18 to 19. We broke them up into 2 different categories. First one being fishery independent. Those fishery independent came from Clemson, Auburn, and Alabama Marine Resources Division. And it's important to note that we have individuals throughout our entire study period from the Delta, Upper Bay, and Lower Bay. Our next category was fishery dependent. And these individuals come from creel surveys, commercial fish houses, and fishing tournaments. And these had unknown location of collection, which we tried to assign locality using stable isotopes. So Henry gave a good intro into the otolith processing yesterday, but we took our otoliths, sectioned them to reveal the core and annuli. We aged them using 2 to 3 readers, and then used LA ICP-MS to determine the elemental signature of these otoliths, and then from there, assign them into age-specific and lifetime-specific contingency classifications. So here's the raw strontium to calcium that you would see out of the otolith. We use that regime shift detector that Henry talked about that finds natural breaks and smooth means across the otolith. And then we determine where does that smooth line fall above or below a 1.1 strontium to calcium ratio. And what that 1.1, as I mentioned earlier, it's only about 1 part per or 1 ppt salinity. So above it would mean that that individual is living in an estuarine habitat. Below would mean they're living in a freshwater habitat. And so we categorize into 90% of the otolith signature. So if 90% was above, we consider them to be estuarine contingent. If 90% was below, we consider them to be a freshwater contingent. If 90% was neither above or below, we determine them to be a transient contingent, meaning these individuals are either moving between fresh and estuarine habitats or residing in an area such as Upper Bay that sees annual fluctuations within salinity. So here's an example of what you would see from an estuarine on top, that transient individual, and then a freshwater on the bottom. For age specific, we did the exact same thing we did for lifetime, just did it from annuli to annuli instead of the entire length of the otolith. Stable isotopes were processed at the University of Hawaii. We processed them for compound specific and bulk isotopic signatures. With what we're looking for bulk, with bulk is the carbon really shows your marine versus terrestrial carbon, where nitrogen looks at that trophic position. And then for stomach content analysis, we just did macroscopic inspection of prey to look at relative prey size, so prey size relative to the flounder that consumed that prey. For our analyses, we were looking at river discharge to see if it could tease out any of these contingent classifications, see if we could really delineate whether those individuals are moving or staying stationary. We used multinomial logistic regression with river discharge and age 0 contingent patterns. And then we use quadratic discriminant analysis to assign locality of collection for our fishery dependent samples using leave one out cross-validation. For our growth, we use back-calculated growth using the Dali method, using linear effects models in R, having random effects of sex, cohort, and age of capture. And then we did a very similar thing for condition, just looking at log length and log weight. So getting into our results, this figure has a lot into it, but if you look at the columns from left to right, you see we broke it down into region. So the delta on your left, Upper Bay, and then Lower Bay as you move from left to right. And then our contingents are from the bottom up, so estuarine on the bottom, transient in the middle, freshwater up top. And the actual figures themselves are length frequencies for those contingents and regions. And then the shading within the pie charts is darker is freshwater, and as you move to lighter gray, you get into that estuarine contingent. So overall, from our fishery independent samples, you see a large proportion of them are actually freshwater contingents, and then transients being the second most, and then estuarines being actually the least likely of our contingent types. When you look at stable isotopes, we were able to accurately assign our location of collection with a 90% accuracy. And so what you'll see in these two figures, they both show the exact same data. The figure on the left is just the raw carbon to nitrogen, where the figure on your right is a principal component analysis with a center-weighted centroid. But with this, you see a distinct separation between delta and lower bay individuals. This is largely driven by carbon, that at about 24C, you transition from terrestrial carbon to marine carbon. But with this, you do see some overlap with the Upper Bay. Predominantly, though, Upper Bay seems to be aligning mostly with the delta individuals. So now this is isotopes again, but we broke it down by contingent instead of region. And here you see a lot of overlap between those transient individuals, showing that these transients are likely residing in the exact same habitat that we see for these freshwater and estuarine individuals. So this is the same figure you saw from earlier, except we added a column on the far end, and this is our fishery dependent samples. And with fishery dependent, you see that we have almost a complete opposite from what we saw from our fishery independent samples, with only 4% of them being freshwater residents or contingents, and that 58% being transient. And so a reason you may be seeing this increased transient is those may have been freshwater individuals previously and then moved into saline habitats before being harvested. And then I wanted to remind you of our fishery independent data, so that way you can see the the stark differences. But with this, we wanted to see if we could assign locality of harvest or residency for these fishery-dependent samples. We use the Fisher's exact test to see if there's any correlation between the patterns we're seeing with fishery-independent with any of our regions or contingents. And we saw that there's actually a significant difference between all of them, saying that these fishery-dependent samples are actually likely coming from each of these regions, and not one particular region seems to be contributing most to our fishery. So this is the quadratic discriminant analysis that we use for our fishery-dependent data. You can see the triangles are the fishery-dependent, the circle are fishery-independent. Our quadratic discriminant analysis assigned them into 2 of our 3 regions, saying that 29% likely came from the upper bay and 71% came from the lower bay. It's important to note within this, though, we have 8 individuals that have a distinctly different isotopic signature from what we're seeing within the rest of Lower Bay. 3 of these 8 individuals we actually know were harvested in a tidally influenced lagoon called Little Lagoon that is not directly impacted by the river discharge that we see in Mobile Bay. So it's important to note that if you're going to try and use isotopes to develop some sort of residency or comparison using otolith chemistry, that you need to incorporate the entire isoscape of that region that you're wanting to compare. So this is looking at our river discharge analysis. As I mentioned earlier, we were trying to test the hypothesis that these transient individuals are actually showing lack of movement, not actually moving across estuaries. So we use the age 0 proportions with the river discharge. River discharge increases as you move from left to right on the figure, and they are broken down into the estuarine residency on top, freshwater in the middle, transient on the bottom, and the lines themselves are actually the region of collection. And so with this, you see increased proportions of freshwater and decreasing proportions of transient individuals with increasing overall discharge. And we believe this is actually strong evidence to suggest that these individuals are residing in an area where they are seeing seasonal fluctuations of salinity and not necessarily they themselves are moving across estuarine habitats. Now, when we look at our growth analysis, we see that there's actually a significant difference between growth for our different contingents, with estuarine and transient contingents having significantly higher growth rates in the age 0 and age 1 growth, which is your top 2 figures. And then by the time they reach age 2, there seems to be a uniform growth throughout. You look at contingents, and you see very similar patterns in that those estuarine contingents seem to be residing at seem to be having a much higher condition at that age 0 and age 1 growth, where by the time they reach age 2, again, we see a uniform throughout. Now, when you look at trophic position, the top 2 figures show trophic position. The top left figure is using compound specific, which is generally considered your more accurate because it accounts for the base level nitrogen within the ecosystem, where the top right figure is your bulk nitrogen value. The figure on the bottom is that prey relative size. So as I mentioned, it is prey size relative to the flounder that consumed that prey. But with each of these, you see very similar results to what you saw with our fishery, or similar results that we saw with the condition, in that you see that elevated trophic position, meaning these individuals are probably foraging on larger prey at higher trophic positions within the estuary. So in conclusion, we feel like we have strong evidence to suggest these individuals are staying relatively stationary during that early growth phase. We see that a lot of the fishery-dependent samples are actually coming from transient and estuarine contingents, even though we saw a lot of freshwater within fishery-independent samples. And we see that trophic position growth and condition are all highest at our estuarine, at least during early life growth. So to compare our results across the Gulf of Mexico, the top 2 studies were done in Alabama, the bottom was done in Texas. You see that transients seem to be really uniform across the entire Gulf of Mexico. However, there seems to be a high variation between your polar opposite contingents. And this may be due to the fact that Alabama is a river-dominated estuary and has a lot more influx of fresh water than we see in those Texas estuaries. But with that, we'd like to see more research looking into these contingents across the entire Gulf of Mexico, see if we can really understand more about how these contingents are acting in different freshwater influxes, as well as see if there's any sort of genetic differences between these contingents, as we've heard lots of talk lately about them potentially spawning in different locations. With that, I'd like to thank our funding agency of Alabama Marine Resources, as well as all the universities and labs that have helped us throughout the time of our study. And with that, thank you, and I'll take any questions.
Mike, we have lots of time for questions.
Nice talk, Jared. I really enjoyed that. I had a quick question about the nitrogen and the trophic position. Was there any differences in the baseline nitrogen between the upper bay and lower bay that might account for some of those differences outside of prey?
There was a little bit, especially when as you saw with the Little Lagoon, it definitely had a much lower baseline nitrogen, but we tried to account for that using those compound specific. But yes, there was slightly different baseline.
Nice job, Jared. Henry with Auburn again. Have you looked at all at that sort of homing behavior that we've seen, where maybe fish are moving out away from freshwater habitats and then coming back into it later in life? Like, have you looked at those striding profiles to see whether there are like annual patterns in striding changes, or with the lifetime of the individual, or was it just if it was bouncing around that threshold, it was a transient fish?
We did look at age-specific relationships, and for the most part, they all either stayed within their contingent for about the first 2 years. We had 2 individuals that moved from what was classified as estuarine into freshwater, but for the most part, we did see what is considered like the putative life history, in which they had that lower freshwater early in life, and then as they became older, they moved into estuarine habitats.
Yes, this is Fred Scharf from North Carolina. Nice talk. I wondered if, if you thought that the combination of using the stable isotopes, particularly the carbon, in combination with some of the trace element work that you guys have done, would, if you looked at those things together, would it give you sort of more resolving power in terms of identifying, transient and resident fish over periods of time?
I think it would. I think that as I mentioned earlier, you have to have that entire isoscape to really tease out the nuances that we're seeing with this. But yeah, I think if you're able to collect flounder across the entire region of your study, you would be able to maybe delineate more on these transients, especially with stable isotopes really being representative of about the last 6 months of life. So you kind of get that end of life where otolith tells you more the entire life history of that individual.
Hey, Jared, Kenneth Erickson. Was the fishery dependent effort, do you know, like the samples you got, were they pretty evenly split between your 3 different regions, or was the fishing effort more focused on the lower bay area?
Unfortunately, we have unknown effort on that. I would say, based off of what we're seeing predominantly within Alabama's fishery, it doesn't seem like the delta or freshwater is highly targeted for flounder. So I think most of your effort is coming from that upper and lower bay, which is where our QDA assigned that location of collection. Okay, thank you.
Thank you, Jared.
Thank you, Jared. For our next talk today, we'll hear from Fred Scharf, and we'll talk about scales of movement along the U.S. Atlantic coast inferred from conventional electronic and natural tags.
use this time. But I won't hold it up here. I'll try to hold it down here. Thanks, Mike. So what I was hoping to do with this talk was give you guys a summary of some of the work that we've done in North Carolina that dates back to the mid part of the 2000s, both in terms of conventional tags, ultrasonic telemetry tags, and at different scales of movement, both sort of fine scale and broad scale. And so you can see sort of hardened sort of migration triangle up here, and trying to sort of connect this complex life history of southern flounder that we've been talking about so much with some of the dimensionality of this. And we've looked at a lot of these movement questions mainly because it's so essential for management in terms of, you know, protection of nursery habitats in the estuary, identification of these areas where they aggregate, the potential even for temporal closures during the migratory season. We've seen that executed recently in some states, and as well as the scale of offshore movement and mixing among states to inform the spatial scale of management. So I'll start in the estuary in terms of some finer scale habitat use and migratory dynamics. And the previous past tag return studies using conventional tags had provided some evidence for lots of things, some of which we've heard about in some of the other talks, you know, related to sort of within limited movement in the estuary, possibility of filipatry, right? Some size dependence in the migration timing, and overwintering of immature fish in the estuary. The problem with the conventional tags, right, is that the inferences are limited by the lack of continuous data that we have for the fish. We get the release and the endpoint, and that's it. And so we started a broad movement study, right? Our poster said movement study. We were really focused on mortality, both fishing and natural. But if you say you're looking at fishing mortality, the fishermen don't tend to want to cooperate. So we don't mention anything about fishing mortality in our posters and advertisements. We just say we want to know about their movement. Right? And so we did this using acoustic tags, V9, similar to the ones that other folks have talked about. And we did this in the New River estuary. And this is a estuary along the south central coast of North Carolina, moderate size. You can see the receiver array that we had positioned throughout. And we did this at multiple spatial and temporal scales. We were able to identify lots of information about movement, emigration, which I'll talk about next, and mortality, which I talked about a little bit yesterday. We did focus a little bit up here on this major tributary up here. And this tributary is about 4 or 5 kilometers in length from the mouth to the headwaters of where we can navigate any further. And we did a lot of extensive sort of passive listening in here and active listening in here to look at some fine scale movement. And the first efforts to look at that fine scale movement was a PhD student, Lisa Hollenseed, who did some focal follows where she actually followed individual fish for periods of 24 to 36 hours and tracked their positions every 10 minutes. And this was cheaper to have her do it than to buy a VPS system, right? And so you can see these are the movements of individual fish. Sometimes she followed 2 fish in a given focal follow that were close together, and so she tracked both of them. And the colors just represent time from the beginning to the end. So you can see some movement of this fish maybe in this direction. But notice the scale bar in each case is only 50 meters. And so over the course of a day or a day and a half or so, these fish are not moving in an area that's too much bigger than the size of this room, maybe twice the size of this room. And she did a lot of this, right? So she was sort of sleeping, and in between she'd sleep for 8 minutes and then listen for 2 minutes and then sleep for 8 minutes. And she'd have one partner with her on the boat who took this picture while she was sleeping. We also did, so beyond the fine scale sort of daily work, is we were looking at weekly movement patterns within this creek. So we had that passive array, we complemented that with some active listening. And one of the things that we found over the course of the study was that the flounder aggregated some really, in these predictable locations, sort of in the mid part of the system. We also found that they displayed, similar to that daily activity, which was low, they displayed very low activity throughout the summer and early fall season. So many individuals remain sort of within 1 by 1 kilometer spaces from months at a time. And so we wanted to also look at whether there was anything unique about these places, and these are a couple plots of prey species richness and prey species catch per unit effort. So we surveyed some of the potential fish prey that we were seeing in their diets, and where the flounder overlap tended to align pretty closely with where the prey fish overlapped. And we also saw some evidence of sediment type, as well as sediment organics, and maybe a moderate influence of flow dynamics, tidal influence within the creek. At a broader scale, Lisa looked at some fishery independent data, whole estuary scale fishery independent data, and essentially found what we'd expect, at that scale in terms of the importance of depth, as well as distance from shore, which are highly correlated with each other. But basically, they were found pretty close to the shorelines within a couple hundred meters, and mostly at depths of 1 to 2 meters. And she also found some associations with some substrate types and an avoidance of places with man-made structures, docks, pilings, and those sorts of things. So after we spent some time sort of learning and thinking about what we were seeing in terms of their movements and things that were associated with those habitat associations for southern flounder, we started to look at migration dynamics, because in order to estimate mortality rates, we had to be able to separate out emigration from the system from natural death or fishing death. And so we wanted to look at emigration rate routes, the temporal window, and how consistent those routes were in the windows, just like some of the talks we've seen this morning in the Gulf states. And then we looked at factors that might trigger this migratory behavior, and looked at multiple factors on different temporal scales using some time to event models. And we did this again in this system. And the nice thing about this system is that it has some natural migration corridors, sort of pinch points, if you will, where the estuary narrows. And it was very easy for us to put, multiple gates in there, build some redundancy, and identify, the movements and timing of fish. And it also allowed us to estimate our detection efficiency really carefully throughout each of these locations in the system. So what did we find in terms of emigration timing? Well, we found that it was really consistent and predictable. So about 80% of the emigration in North Carolina occurred between mid-October and mid-November. So maybe about a month earlier than we tend to see it in the Gulf of Mexico. So it's more concentrated in late October as opposed to mid or late November, like we've seen in Texas or Alabama. It gets cooler there earlier, right? As you'd expect, we're at the northern edge of the range. And so not only was the emigration really consistent, you see a few fish go out a little earlier, and then a little bit of a longer tail to the end where you get some emigration trickling into December. It was also really rapid. So when the fish began directed movement out of the system, they left within a week or 10 days, most of them. Right? A few fish would take longer, but most of the time when they decided to go, they went very quickly. And so this was, because the mortality study was extensive and occurred over a 5-year period, we had 5 years of emigration data from this system, which is really nice. And so this is just a Kaplan-Meier curve fit to residence probability as a function of date. And what you see here is that you see that window between sort of mid-October and mid-November, and the estimate of 50% residence probability was just before Halloween. And we did see a little bit of year-to-year variability, but you can see it's not extensive, right? Most of the fish are still leaving within that same window of time, but we did see one of the years in the study between where they left, they tended to be a little earlier within the window, and another year where they tended to be a little bit later within the window, and then 3 years where they were sort of right down the center of that window. And in terms of the factors that contributed to some of this variability in timing, right? We built some time to event models, using some proportional hazard models on different temporal resolutions. And we looked at a lot of forcing variables in terms of temperature and wind, barometric pressure, and precipitation. You know, as just like here, we're about to experience one in a little while, right? The North Carolina coast, especially in the fall, is characterized by the passage of these cold fronts. And sort of contrary to what most of the fishermen would say, you know, in terms of, you know, you get a good blow and then you see some fish moving, right? The data we had for 5 years in New River suggested that the response was due to cumulative changes rather than very abrupt shifts. So the most important predictor variables in terms of initiating movement, now this wasn't their exit date, it was the date when they started directed movement south. They were cumulative indices of precipitation over the past 1 or 2 weeks. So it was almost the passage of multiple fronts that got fish moving rather than any single front. But we did need, because of the consistency that we saw, we didn't have a lot of contrast in terms of the timing of those movements. So if we had a little bit more contrast, we think we could tease those environmental factors apart a little bit better. So I mentioned this in my talk yesterday, obviously, about the mixing and where do they go when they migrate. And so we have several pieces of evidence to inform that in terms of their movements. And this all started for us way back in the mid-2000s when we started a large tag return study. Again, this was in a focused effort to estimate harvest rates, right? But we learned a lot more than harvest rates as part of this study, and the movements was one of them. And so we did this as a companion study with a colleague of ours, Kevin Craig, who did work up in the Neuse River up here, and we were working in that same system in the Neuse. We released 4,000 fish with these cinch-up tags that are positioned in the caudal peduncle, and we released them in 2 consecutive years to estimate harvest rates, but we got a lot of information about movement. In addition to that, right, so this in the green here are our tag numbers from the Neuse and the New River, right? And then we have these other numbers are historical tagging that were done by the staff at the Division of Marine Fisheries. Some of this happened in the 80s and some of it throughout the 90s. And so we were able to use their historic database to also inform what we were seeing in the mid-2000s. And as I showed yesterday, that we see, again, the conventional and acoustic tagging data that we just talked about in the estuary shows very sedentary movement, very small activity spaces, even seasonally, of less than a half a kilometer squared. But then we see this extensive migratory movement, you know, 20 up to hundreds of kilometers, right? And all of the southern flounder that we tagged in the North Carolina system and the ones that the division had tagged in the 80s and 90s, all of the recaptures of those conventional tags occurred to the south. You know, as far south as the east coast of Florida, lots in Georgia, South Carolina. You know, one interesting thing that we noted is they like Myrtle Beach, right? You know, I think like most people, right? They like Myrtle Beach. And so we saw there's a lot of fishing pressure right around the border, around Little River. So there's a heavy recreational fishing presence in this area. So we got a lot of recaptures here. Anecdotally, with our acoustic tags, we also got a lot of recaptures to the south. One thing to note that I didn't note in terms of our migration is that we didn't see in those 5 years, we saw no evidence of filipatry. We never had an acoustic tag that was a confirmed emigrant re-enter that New River in 5 years. So we never saw it once. So all the large-scale movements to the south. The other piece of information that I mentioned yesterday that helped us to look at connectivity and movement was looking at otolith microchemistry and looking to see where adult southern flounder were sourced from was really our major question, right? And so we evaluated the chemical signatures during the juvenile period. So we isolated this period, built a nursery atlas, fish from North Carolina, South Carolina, and Florida. We weren't able to get fish from Georgia. And then we took older fish, 3- and 4-year-olds as adults, and then looked at that same period to assign those fish back. And as I showed yesterday, the nursery origins suggested a lot of movement among states where the adults captured in North Carolina at 3 and 4 years old were sourced to all 3 of the states in terms of their nursery origins, and similarly for South Carolina, right? Almost evenly mixed, which again, it suggested that this was at least partially driven by post-spawning movements of adults. Wherever they spawned, they may return back to different states. And so to finish up, I just want to acknowledge those particular students that helped with this. Will Smith back in the mid-2000s. Trevor was focused on the mortality estimation, and then Lisa's work was all about the movement and migration work. And the fact that some of that migration work, you might say, well, that's 5 years of migration data. How come that's not published, right? And some of that's my fault, right? Maybe a lot of it's my fault. But Lisa currently, she finished her PhD, and she currently works for the Gulf of Mexico Fishery Management Council, right, as a biologist. So some of you may interact with her. So I would appreciate it if you would nudge her occasionally and say, wow, we saw this great migration data. It would be great to see that in the literature, Lisa. That would help me. All right. With that, I'll take any questions.
Fred, nice talk. I enjoyed that. I was curious just if you could talk about a little bit about your conventional tagging where everything seemed to be moving in a southerly direction, but then your otolith chemistry data, which indicated there's some movement maybe back north into North Carolina from southern parts. So it was interesting the different inferences from different tagging approaches.
Yeah, most of the recaptures from the conventional tagging that occurred in the southern states tended to occur within 6 months to a year of that release. So a lot of those tags were like summer and fall. Fish may have left North Carolina, and then those recaptures occurred the next spring or the next summer. And so it suggested to us, okay, there's some southward movement to spawn, and when those fish are coming back inshore, they're coming back inshore to the south. And then when we looked at Verina's data, which was fish that were 3 or 4 years old, and we saw them maybe being sourced to some of those other nurseries, that led us to say, well, maybe they move around more after that than we thought, right? So maybe there's this initial sort of slide to the south for spawning, and yet, after that, when they're 3 or 4 years old, they may get more sort of rambunctious about where they go, but. We were hoping to learn more about that from the satellite tags. I'm trying to set Shelby and those guys up. And there's some interesting stories with that as well. What's interesting is not only is that North Carolina data that I showed, but in that Craig et al. Paper, we also summarized the data from Charlie Wenner's work in South Carolina. He did a lot of tagging in the late 80s and early 90s. There's also a paper from Georgia from the 80s, a Musick and Pafford paper, where they released a lot of fish. And in those 2 states, the fish that moved extensive distances were also recaptured to the south. So a lot of the South Carolina fish showed up in Georgia. A lot of the Georgia fish showed up in Florida. There was one fish in Georgia that showed up in North Carolina, but it was a really small individual. We suspected it may have been a male when he tagged it. And so the males may do something very different.
Thanks.
Mike Dance, LSU. Nice talk, Fred. I had a quick question. I think I know the answer, but I just wanted to make sure that southward movement, is that with the longshore current or the predominant direction?
That's a good question. I don't know. Okay. Yeah. Mike's not here. Do you know? Okay. I don't know. I think not, but I think it's the opposite. But typically, right, is moving up the coast, but I don't know.
Hey, Henry with Auburn. Maybe the southerly movement of adults is to compensate for northern larval drift on that current. So maybe they're moving counter current, and then that's why you're seeing that northern dispersal or fish that are captured in one estuary, or fish that are captured further north than their natal origin.
Yeah, I mean, that's what we always presumed, right? Because we, the ichthyoplankton data and the lack of fish inshore during those winter diving surveys suggested that the spawning was happening closer to the shelf break, right? Out near the Gulf Stream front. And so we always figured, well, that southward movement was because they're dumping their eggs in a place where the current's ripping really fast to the north and east. And so, when we first looked at a lot of that conventional tagging data, we said there must be a big aggregation sort of in that armpit, between like right in that crux between Georgia and Florida, like right in that northern area, and they're releasing a lot of eggs there, and then they're drifting up and then spinning off with eddies and other cross shelf fronts, and the Walsh work suggested that the flounder larvae were at mid-level depths, which tended to have net shoreward transport, and they found smaller larvae near the edge of the shelf and then bigger ones as they got closer to the inner shelf. That was across the North Carolina shelf. So we kind of assumed that was happening along the Georgia, South Carolina, North Carolina coast, that the eggs and larvae were being dropped down near the bottom. And then as this conveyor belt shifted them up, they were spinning off to the left. But the satellite tag data doesn't really support that conclusion, so I'm not really sure.
Thanks, Fred. For our next talk, we'll hear from Shelby White, and she's going to speak to us about assessing migration and spawning habitats of southern flounder using satellite tags.
Thank you. All right. Okay. So yeah, I'm Shelby White. I'm with the North Carolina Division of Marine Fisheries, and today I'm going to give you an update on a collaborative project between the Division of Marine Fisheries and the University of North Carolina, Wilmington on some satellite tags that we attached to southern flounder. This research was funded by the Coastal Recreational Fishing License Fund and the Commercial Fishing Resource Fund, very briefly, both of which just use license sales to fund relevant projects. Okay, so a lot of this has been covered, but I just want to touch on it real quick. There's a lot of missing pieces of the puzzle for flounder. We know a lot of things. We have evidence of long offshore migrations. We have evidence that some of these adult fish may even spend time offshore based on dive surveys and recreational catches. We have some evidence from ichthyoplankton surveys that they may be spawning near the continental shelf, and that there's evidence of the southerly movement like Dr. Scharf just said. But what we're missing is what these flounder do after they spawn. Where do they go? Does spawning occur when they leave from these estuaries and select locations, or do they go adjacent to the state from which they left? And then the single stock approach, we don't know really how they're mixing, when they're mixing, or where they're mixing. And then the habitats that these flounder are using before, during, and after spawning. So hopefully some of this research will give some insight into that. And so the objectives of this were to identify potential offshore spawning areas used by southern flounder, identify movements prior to and after the winter spawning, and identify migration corridors and offshore habitat preferences of southern flounder. And so to introduce you to the tag type that we used two models of the MRPAT and MiniPAT. Both are from a company called Wildlife Computers based in Seattle, Washington. They detach on a predetermined date. So you can set the date, say I want the tag to float to the surface. It burns through a copper wire, floats up, transmits to the satellite. And these tags, particularly the MiniPAT up top, give us release location and min-max temperature range for the time that they're in the water. Now the MiniPATs, which look almost exactly similar, have the same characteristics, provide additional data on depth and temperature at specified intervals. And you can see both of these tags are pretty large. So this is a novel way to tag southern flounder, which meant that we had to pick bigger flounder so that these tags didn't interfere with swimming or any of their lifestyles. that they have or their characteristics. So all fish tagged were 500 millimeters or greater, which translates to about 19.5 inches. And then also we can kind of improve our success rate of tagging females with the larger fish since flounder are sexually dimorphic. And all tags, or all fish that we use, also received a high reward spaghetti tag. So fishermen have a $100 incentive to call and return the tag. And this is just an additional piece of information that we can get if anglers report their tags. So some tagging methods, very briefly. We conducted some tank trials to determine which tagging methodology had the highest retention and the fastest healing rates are the best healing rates. So we didn't want to put these fish or these tags, which are pretty costly, in the water and have them float away and not be able to collect any data. So what we did is take the spaghetti tag, which is yellow right there, and if you're not familiar with it, it really just looks like a short spaghetti noodle. Attach it through. It goes through the fish on the underside, and then we put a blue disc to distribute that pressure and prevent the tag from pulling through. So before we had the blue disc, we noted some instances of kind of wear and tear where that flounder was on the bottom, and we didn't want to have tag retention be an issue. And then it comes back up, you tie the spaghetti tag on. That orange piece, which is where it disconnects from and burns off from, stays attached to the fish, and the black part of the tag floats to the surface. And then you can see near the caudal peduncle, the high reward spaghetti tag that once again could provide us an additional piece of information if fishermen returned it. So some tagging method specifics. This was a 2-year tagging project so far. So we started in 2020 and then tagged again in 2021. And so you learn a lot in the first year. Like I said, this is kind of a novel way to tag southern flounder. So in the first year, we used 3 tagging locations and put out 130 tags between Marine Fisheries and UNC Wilmington. We had the satellite set to communicate within 12 hours. So we found that maybe that wasn't the best method because that means it has to stay dry for 12 hours. And when it's floating around in a volatile ocean environment, hopefully if it's offshore, that's a pretty hard feat to accomplish, especially if you have algae and other things growing on it. And the tags were released at 15-day intervals starting on January 1st all the way to March 15th. So during the suspected spawning period, every 15 days we'd have a subset of tags released, float to the surface, and communicate with us. In year 2, we switched the tagging locations a little bit, and it's actually 5 tagging locations. I had one of them combined to release 94 tags between both groups. Satellite communication was changed to 8 hours. This seems to improve our delayed communication. Any ghost tags that we had a lot of success with changing that. And we adjusted the release times for randomness. So maybe we can get a better idea of when they're popping off, their release location, and a more, we don't want to limit them to those 15-day intervals, assuming that all fish behave the same. We also painted them with anti-fouling paint, and that seems to have helped the communication as well. So a good lesson for anyone that has satellite tags. And then we actually extended collaboration with South Carolina and Georgia in year 2. So we are extremely appreciative of their work and their help with this. And they did a great job of looking for fish. Georgia was not able to find any in year 2, only because we may need to adjust the time that we send them so that when they see the larger fish, we're able to get some tags out. So, tagging locations, just to go through year 1, like I said, 130 tags, all distributed in North Carolina. So we tagged as far north as Albemarle Sound, that little piece up there, Hatteras, and then Core Sound. And then in year 2, we switched a little bit, did not tag in Albemarle Sound, but kept our Hatteras site, our Core Sound site, Masonboro, Little River, and then in Ashley and Combahee Rivers in Charleston, or near Charleston, so I put them together, and that was South Carolina's tagging effort. So in year 1, we used only pound nets for our sampling. We worked with some commercial fishermen that were willing to help us, and in year 2, we went back to the same commercial fishermen, but also expanded the efforts to a gill net fisherman and using some independent gear sampling as well. So when we get these tags back, it's a lot to get. You get some location data, and you get a lot of spreadsheets, a lot of zip files to go through. But what it really gives you is a location that's within one of these accuracies. So what you want to see is a numeric value, a location where 3 is the best, and then go into 1. The alphabetical locations don't really give you a good accurate error of estimation. So we cleaned the data following some methods in the literature. So within the first 24 hours, if a tag uploads, we went to that data set all within one day and looked for that 2 hours after the first communication with the satellite. We deemed that those were pretty good representations of what we were looking for, and we picked the first 3 that occurred within those 2 hours, and if there was no 3, we picked a 2 or a 1. So we omitted any A, B's, or C's and zeros from the analysis, and so that left us with about 145 tags out of 224 that communicated across both years, which is right in line with what the literature is seeing. So year 1 and 2 combined tag and releases, time for the exciting part. So they're color-coded by the place that they were tagged, so Admiral Hatteras, Core, Sound, Masonboro, Little River, and Charleston. So you can see there's a whole lot going on, right? We wanted all the tags to be offshore, and then we knew exactly where they were going, but that is not the case. So they are scattered throughout. We do see some evidence of offshore movement, but we also see a lot of fish inshore, and it's not really dependent on where they were tagged, how they were tagged. And then, even the South Carolina tag went to that kind of inner shelf environment. So what we did was we looked at this a little bit deeper, right? We wanted to know what the difference between an inshore fish release and inner shelf fish release, which we kind of consider to be right in this line, and then the outer shelf or that continental shelf where we suspect that they're spawning. So what we did was we measured the distance from the nearest land point that the fish was, and then put that distance from the shelf to the land. If it was over 50% to the shelf, it was considered an outer shelf fish. If it was inside that 50%, it was an inner shelf fish or near shore fish. And of course, inshore, we're inside the estuaries. And so the largest percentage of fish were inshore and the inner shelf environments. We did get some fish, once again, that went offshore, but 10% of them. But what we're seeing, or maybe what we're not seeing, are a lot of fish traveling those long distances south. And that's not to say that they don't travel south or that they don't mix or anything, but we're just not seeing it during the suspected spawning period from these adult female southern flounder. So that could lend to it being the males. There's a few questions as far as why are they remaining inshore. Maybe it's something to do with maturity more than it is skipping spawning, or maybe it is due to skipping spawning. The nearshore environments that they popped off in, are they staging there, waiting for the perfect condition to go offshore? Are they deciding whether it's a good year to spawn, or maybe not so much? And in addition to that, we are going to look at some temperature data. So like I said, the mini pads and the MR pads both provide temperature data on different scales. So from the mini pads, this is what we get. We get temperature across the time the fish was in the water, and we're thinking maybe we can classify if they were in inshore, inner shelf, or outer shelf environments just by temperature variability. So when the temperature is really variable, that can suggest that a fish is in inshore estuarine waters. When we get temperatures that get a little less variable, but it still exists, maybe those are the inner shelf waters where there's still have some movement of temperatures throughout the day. And then maybe an offshore environment is those really tight temperature ranges that don't move too much, and we can maybe suggest that the fish is offshore. And so we also did some data analysis. This is a little bit preliminary. We have some more that we want to explore with this, but we use multinomial logistic models using December year 1 and inshore as their baseline reference to compare everything to. And in year 1, we found effects of tagging location that we did not see in year 2. And that is likely just because we put a little more tags out in certain areas in year 1. And then in year 2, we had more fish inshore. And so we expect that because maybe we missed this spawning period because our tags actually stayed on. They were painted with anti-fouling paint. We didn't have, the fish was tracked throughout its entire movement. And we really don't know how long they spend offshore to know if we missed it or did not miss it. But there's no significant effect between some of the months. And we're thinking that that's just because of the way the tags performed. So real quick, I just want to hit on the mini pad satellite tags. We have put out 8 this year with 5 preliminary pop-offs. The rest are coming off on April 1st. And we put those out in Hatteras and Core Sound. So just want to focus on one of these tags. And maybe, yes. So the tag gives us the depth and the tag gives us the temperature. And what I want to point out, I guess in the gray figure, is the variability between the temperature and depth range. But where those orange arrows are pointing, I want to direct you there. Those fish went really deep, came back up to more surface level areas, and then went back down to about the same depth, all within a 20-minute time span. So we're thinking that maybe this is evidence of some sort of batch spawning going on once they get offshore. And just for reference, another figure where you see the exact same sort of movement of that may potentially indicative of batch spawning behavior. And so looking forward to the future, what has happened in next year, we're going to expand our tagging efforts. So hopefully South Carolina and Georgia are willing to help us again and potentially purchase additional mini-pat tags so that we can back trace those with the MR-pat tags and look at some temperature variance and potential locations. And then just explore additional models with years as random effects and things that we know would be more statistically useful. And so thank you to all the commercial fishermen that helped us, to Mason Collins, Dr. Scharf, and Mark With and Mike Leffler, who have all been a great part of this project, and we've all worked together really well. To the collaborators, South Carolina and Georgia, and to the staff that helped get all these tags out. It was not an easy task. So any questions?
Great talk, thank you. This is Steve at LSU. My question was, I think if I caught it correctly, you were tagging fish at 19 inches and larger?
Yes, 19.5 inches or larger.
So I'm just thinking, at a pretty large size, many of those fish may have been mature in a previous year, and I'm wondering if there's any chance that fish that are entering into their first year of maturity may do something different than fish that are repeat spawners. And I'm not really sure what my question is, other than to say, might there be something going on that you're detecting that doesn't describe the fish that are entering their first maturity event or maturity season?
Yeah, I would say that's definitely possible. Of course, like I said, we made this figure and fish were all over the place. So we were like, what is actually happening? And there's a few questions that, this is really just the start. It's fun research, right? You get a research project, get some answers, and then it leads to a whole lot more questions. So yeah, that definitely could be something going on.
Really interesting talk. So John Godwin, NC State. I wonder, those rapid ascents, you speculate maybe spawning events. I was wondering if the timing in between might match what we'd expect for sort of sequential spawns.
I guess from the literature, anecdotally, yes, that's what we're thinking is happening. That's, I guess, the next question, right? We don't really know how long they've been out there, how fast things are, but we know that a lot of flatfish species have those moving to not shallower waters, but up more near the surface waters and going back down and doing this kind of batch spawning behavior. So that is what led us to think that potentially it could be some batch spawning going on.
Hey, Eddie Leonard with Georgia DNR. Did you have any or sense of whether the tag itself might have restricted their maximum range of movement? Whether the carrying that tag might have altered how far they were able to.
Yeah, so hopefully not. We put them in tank trials for a while to kind of look at their behavior. Of course, the tanks are nowhere near as big as the open ocean. And we didn't see any weird movement from them there, any way they got under structure or anything like that. And we added structure to some of the tanks. Since we've been talking about dolphins all day, we were more worried about things like that because the tags are positively buoyant, so it kind of makes the flounder look like a big old target to play with or to eat. And we do think that had happened in some cases, that maybe they were prey or foraged on. But as far as large-scale behavior changes, that not exactly sure, but we don't believe so.
Mike Dance at LSU, great talk. What on the Mr. Pat tags that popped off in the nearshore zone? What What kind of depth was that in? Did you have like kind of? I might have missed that if that.
So we don't get depth with the MR-PAT tags.
Yeah. So I guess maybe my question is, have you looked at that? I guess, and you may have just answered that in terms of at the pop-off locations, the depth. Taking bathymetry from the nearshore zone. What kind of depth range were they popping off at?
So that is the upcoming part of this research. We want to overlay, I should have said that, overlay this with some of the structure between those kind of inner shelf, near shore environments and the outer shelf. And in doing so, we can see if they're using, artificial reef habitat or what the depth is looking like or what the bottom structure is looking like. So that is to be determined and coming up soon.
One of the YouTube viewers texted me and said, "OMG, look at the size of that slab." So they were impressed.
Good.
Dylan Kiene at South Alabama. Great talk, and that was an incredible job pulling that off. I was curious about if there was any variability in the timing that you were detecting those fish on that outer shelf, and what time of year that those were being detected out there.
Yeah, so that is why we ran the odds ratio in our logit models to see if there was any significant difference between the timing, because we actually had a figure at one point that showed the month that it popped off, and it was more in that January, maybe some February, December. Sort of things, but when you look at it through, a statistical analysis, there is no difference between the months. So they have the same kind of chance of being inshore or offshore or near shore during those times.
Cool. Thank you.
Yeah.
Thank you.
Yep. Thank you.
Thank you, Shelby. We have one more talk before lunch, and it'll be Caitlin McGarigal. Did I say that correct? Oh, sorry. Identifying the spawning and offshore migration corridors used by the North Carolina stock of Southern Flounder.
Thank you. You're pretty close with the pronunciation. It's Caitlin McGarigal. And thank you guys all for sticking it out to the end. That rarely happens at conferences, much less when we have tornado warnings coming through. So again, I'm Caitlin McGarigal. I'm a research associate at East Carolina University. And today I'm going to be talking about some of the research that we're doing, taking a multidimensional approach to looking at Southern Flounder offshore migration corridors and spawning grounds in North Carolina. So the benefit to going last is that everyone's done all the introductory and background work for me, so thank you. I'm just going to touch on a couple things. Obviously, in North Carolina, we're at the northernmost range of Southern Flounder along the East Coast. We've heard before that the North Carolina stock is currently considered overfished and declining. And so this has, important economic impacts in North Carolina because Southern Flounder are one of the most valuable fin fish caught. We've also heard a lot about their life history. So we know that the larvae show up in the estuaries in the late winter, early spring. We know that the adults are migrating offshore in the fall, and we've heard a lot about these sort of gaps in knowledge of the life history. So just to briefly touch on, we don't really know where they're going or how they're using these sort of coastal and offshore habitats in the ocean. We don't know where they're spawning, and we don't know how those larvae are showing up back in these estuaries. In particular, we don't know what proportion of these fish that migrate offshore are then returning to the estuaries, or how many of them are feeding our neighbors to the south in South Carolina and Georgia, as we have seen with some of the conventional tagging data. So this is a highly collaborative project, and there are a number of people involved. So I'm just going to highlight today a couple of the graduate students and some of the work that they're doing. And we have a number of project objectives, so there's a lot of information to cover. So I'm just going to give you the quick highlight reel for each of these objectives. Our first objective was to determine the timing maturity of the fish captured in the estuaries and offshore. And to do this, we worked with commercial pound net fishermen to collect fish in the Albemarle, Pamlico, and Core Sound areas. We also did some independent gillnet sampling in the Albemarle in the second year of our study, as well as some really fun targeted hook and line fishing over the summers in the Pamlico Sound and offshore around the Cape Fear area. Fish were brought back to the lab where they were weighed and measured. One of our master's student, Justin Mitchell, dissected the fish to look at their gonads so that he could identify the sex. He also weighed the gonads to look at GSI, and he's planning to section the ovaries to do some histological staging as well. He also extracted the otoliths, which he section mounted and aged using the annular rings. And he's planning to do some otolith microchemistry to see if he can identify some of these large-scale spatial movements based on the ratios of strontium and barium to calcium over these multiple years. So what he's seen so far, he's dissected 432 fish. Not surprisingly, he found that on average, the females were significantly larger than the males. He did see sort of a geographic separation in their size, with the Albemarle fish being somewhat larger than the core sound fish, which were again larger than the Pamlico Sound fish. Fish that we captured off Cape Fear, they were pretty good in their total length size, but they tended to be a lot skinnier than the fish that we collected from the estuary waters. All the fish that he aged were between 0 and 4 years old, which for males is pretty good distribution across their age, which we know they get to about 5 years. But for females, which age to about 8 or 9 years, we're really only seeing the first half of their life cycle here. When Justin looked at GSI, we found the highest GSI was in late November, and we know that GSIs between 1 and 2 are considered preparing to spawn. So this is pretty consistent with a spawning occurring, or what we hypothesize is spawning occurring in late December or early January. So I mentioned Justin's planning to do some otolith microchemistry. He's still working on that, so I'm going to kind of skip over the second objective and move on to our third objective, which was to determine the timing and migration pathways of these fish as they're leaving the sounds. And we're doing this using some acoustic telemetry. We have an inshore array of 21 acoustic receivers that were deployed across the 7 major inlets in North Carolina, shown here. Our receivers are either deployed from the surface on Coast Guard navigational buoys or from the seafloor on independent moorings. There's also a number of other receivers in this area that are maintained by other groups, and those will show up in later maps as either white or blue sort of pin marks. We all kind of know how acoustic telemetry works, so I'm not going to go into detail about this technology. Although I do want to note that we tried to be strategic in where we place these receivers in the inlets, but some of these inlets are very wide, and so these are by no means considered acoustic gates. There's still a lot of room for fish to sneak out without being detected, and we saw that with our acoustic data. So we, again, worked with those commercial fishermen in our 3 tagging areas. In the first year of the study, we deployed 110 V9 and V13 transmitters pretty evenly between our 3 sites. And so there's a 1-year and 2-year tags respectively. And then in the second year of the study, we deployed another 100 V13 tags. Transmitters were surgically implanted, using standard surgical procedures into the coelomic cavity. And we also tagged fish with an external high-value tail loop tag to sort of incentivize anglers to report recaptures of these individuals if caught later. So to date, we have about 72,000 detections, and we've heard from 79 individuals. We've had recreational anglers report recaptures from an additional 5 fish. Our first surprise was that these fish are showing really high diversity in how they're, migrating through these estuarine waters and how they're accessing the ocean. We kind of naively assumed that they would take the most direct route and leave out of the first inlet that they came to, but clearly they're spending a lot more time using this sort of intercoastal waterway than we had expected to find. We also found that our fish were emigrating later than had previously been reported. So this is just, a simple abacus plot, which we've seen before. And most of our fish are showing up on these inlet receivers in late November, early December, which is later than had been reported of most of these fish migrating through in the October months. But this is probably because we waited until the end of the commercial season to tag fish to give us the best chance that these fish wouldn't get recaught by the commercial fishermen. So we really were tagging the stragglers in the population. So they're just a little bit slower than everybody else. We also were surprised to find that some of these fish were resident throughout the winter, and I guess it's not so surprising after hearing some of the talks today. But we've had 3 fish that were detected moving very consistently around the Barden's Inlet area from December through the February-March time. And I know some people are going to ask, well, were those immature individuals? And we thought about that. But when you look at the size of those 3 fish compared to the other fish that were tagged in the core sound area, they're actually pretty average in size, if not larger than average. So this might be an instance of some of that skip spawning that we've talked about. So if we move on to our 4th objective, which was to track the offshore migration of these fish and locate some potential spawning grounds, we are doing this using an offshore array of acoustic receivers, which are 20 AR units that were deployed at 1 kilometer intervals in kind of this line just south of Cape Lookout Shoals. And we are calling this our acoustic curtain that we kind of deployed trying to look at this movement of fish as they leave the northern inlets and migrate south along the coast. In addition to these fixed location receivers, we also have a mobile receiver, which is on our autonomous liquid robotics wave glider. And we deployed this on 4 10-day missions in December and January of year 1 and year 2 of the study. So overall, we've detected 24 individuals offshore. A number of these individuals we have multiple detections from as they're leaving the estuaries, going through these inlets, and then being detected offshore. So we can start to look at things like rate of movement and other individual movement metrics. This sort of cluster here, that's the acoustic curtain. So it actually did a pretty good job of catching fish as they were moving along the coast here. If we color code these detections based on where those fish were tagged, there wasn't a clear geographic segregation in how these fish are using these different habitats. Although after talking with Patrick at his poster, I have some really cool ideas about how to look at some of these, or using some of these network analyses to look at these, the connectivity of fish using these different habitats. I think it's pretty fair to say that the only consistency with Southern Flounder movement is that they're inconsistent. We've seen a number of these fish make these very clear southern migrations. We've seen some fish move north. We had some fish move offshore out to the edge of the continental shelf, and all of those detections occurred in February. And then we've seen a number of our fish sort of remain in this nearshore coastal habitat, which is about 15 to 20 miles offshore. And this data, this acoustic data is, very similar to what Shelby talked about with the satellite data. So it's nice to see these multiple levels of support with the same movement. We also were excited to find that some of our fish actually returned to the estuaries after being detected offshore. We had 4 fish make this return migration, and they all showed almost identical movement patterns. So I'm just going to show one individual, which was tagged in November, was seen migrating offshore in December and February, and then kind of moving back inshore in early March and back through these inlets by late March. So now we're getting a better idea of where the adults are going and how they're using these offshore habitats. So the next step is to sort of estimate these spawning locations, and we're doing this using some larval dispersal modeling. So the larvae collected for this part of the study were collected through weekly ichthyoplankton sampling through the Beaufort Inlet Ichthyoplankton Sampling Program, which is a long-term monitoring program going back to the late 80s. So any larvae collected through this program were identified to species using morphological characteristics. Southern flounder, their otoliths were extracted and aged using their daily rings. And then one of our PhD students, Brian Bartlett, is using this age to get an estimate of birth date, and then using that birth date to do some backward particle tracking using this connectivity modeling system, which incorporates behavioral parameters and oceanographic parameters to get a probability estimate of origin. So for the oceanographic parameters, he's using this global ocean physical reanalysis database, which provides daily information on oceanographic variables like current and temperature and salinity at the 8-kilometer spatial scale. So that's pretty fine scale when it comes to oceanographic data. So far, I've aged 117 larvae that were collected in the 2020-2021 seasons. Their age ranges, their ages ranged from 15 to 44 days old, and it's not really clear from this distribution whether we're seeing evidence of a single or multiple spawning events. It's possible that fish are also ingressing from different areas. And this is actually supported by the results of Brian's modeling. So here we have a number of maps that Brian's modeling produced. So along the dates along the top are those estimated birth dates, and it's color-coded based on its probability of origin. So just some things to note here are that early in the season, early to mid-January, those probability origins are highlighting those areas in the northern Outer Banks or north of where our sampling occurred in Beaufort. But during the peak larval ingress, the probability or the origin probability is actually south of Beaufort in the southern Onslow Bay area. Although there's a high variability in these probability estimates, and this is very preliminary data from Brian. He's still doing a lot of work on especially that behavioral parameterization of these models. So what would really cinch this whole where is the spawning occurring would be to then go collect eggs and identify these southern flounder eggs in these same areas where we are hypothesizing that these fish are spawning. So to do this, we have been conducting some ichthyoplankton sampling at set stations across the continental shelf. And we actually did all this sampling before we had Brian's modeling results. So if we were to go back and do this again, I would definitely sample in those areas in South Onslow Bay or northern Outer Banks where we think that, or they had the highest probability of where those larvae were originating. But in the first year of the study, we did these ichthyoplankton cruises, sampling 11 stations, stretching out from Beaufort all the way 60 miles offshore to the edge of the shelf. And then in the second year of the study, because of weather, we couldn't get as far off the coast, so we sampled 10 stations that were within that first 30 miles of the shelf. Any eggs that were collected were separated out, and we've seen about 10,000 fish eggs so far that are in the right size range for southern flounder. And then it's going to be the work, or the thesis work, of one of our master's students, Paul Salib, to do some DNA barcoding analyses to identify these southern flounder eggs within that 10,000 eggs that were collected. And he's still working on the methodology for this, so I might direct him to some of you genetics people in the audience. But he's planning to use the CO1 region of the mitochondrial DNA, which has some verified sequence databases that are available and has some established primer sets. So just to bring all these different parts together quickly, we've seen increasing GSI through the fall, which is consistent with that late December, early January spawning period. We've seen high individual variation in their migration routes through these estuarine waters. We've seen some fish overwinter in the estuaries, which we've seen from some other talks as well. Diverse migration behavior in the ocean waters with fish kind of going in every different direction. Although we haven't had any either acoustic or conventional tag returns in areas to the south of North Carolina. So there might be less interstate mixing than we had previously thought. And then again, our preliminary results from our dispersal modeling predict these sort of southward origins for a lot of these larvae that are showing up in the North Carolina estuaries. So with that, I'd like to acknowledge all the ECU team, as well as our collaborators and the commercial fishermen that have worked with us, as well as our funding through the DMF Coastal Recreational Fishing License Fund, and the Matos organization for helping us share some acoustic data with other groups in the area. And with that, I'd be happy to answer any questions.
This is Paul with TPWD. You had mentioned that you had sampled some larvae that were between 15, y'all aged them to 15 to 44 days. Do you happen to have any total lengths associated with those fish, or a general range?
Yeah, so all of the larvae are typically between 9 millimeters and 15 millimeters. I'd say the vast majority are 12 to 14.
Thanks.
But I still have hundreds of larvae still to sample, so. I know that age distribution had a bit of a sample size difference, so hopefully we'll be able to even that out.
Hi, it's Nancy from Southern Miss. So do you guys have any plans or possibilities of actually collecting some of those adult offshore fish to try to get, it's great that you're doing larval and egg work. I mean, that's absolutely fantastic. It'd be really good to get gonads too.
Yeah, gonads from offshore fish. Yeah, that's the dream.
Yeah.
We would have to do probably targeted fish and line, or hook and line, fish and line, hook and line fishing. We've talked a lot about it. It'd be really high effort and low return to do it.
Yeah, I don't know if that off Onslow Bay would be worth it based on your dispersion models.
Yeah, we do know some areas where we could pretty regularly catch fish around the Cape Fear area. So yeah, we might be doing some of that this summer as well.
Hi, Caitlin. Great talk. My name's Kerry Joppy, Texas Parks and Wildlife Department. You mentioned the data sharing group, the acoustic data sharing. I was wondering if you could show me what that was again, and I was wondering if y'all have any communication with the ITAG network that is located southeastern Florida and the Gulf of Mexico.
Yeah, we have not communicated with ITAG. That's definitely something that we should look into. We are mostly part of the ACT group, which is the Atlantic Coastal Telemetry group. But yeah, if anyone has receivers in South Carolina and Georgia, I'd be happy to send you our tag IDs to see if any of our fish are showing up down there.
All right, thank you.
Thank you, Caitlin. That concludes our morning session. So thank you to all of our speakers this morning. If we give a quick round of applause for everyone this morning. And now we have lunch. So same place as yesterday, right, Steve?
Same place as yesterday. If you want to go ahead and start migrating that way, and then we'll check your pattern when we come back here and see if everybody, followed the same path.
And we'll be back here at 1 for the synthesis session, and we'll have the groups split off first. So 1 o'clock.